Hello everyone, I hope you all are doing fine, and now I, Ayushi Agarwal, welcomes you on this amazing platform of Physics Wallah, where today we are going to do the next chapter of Botany, very important chapter from the point of view of NEET, that is anatomy of flowering plants. Now before starting the topic, since morning I am thinking on one quote. The quote which I read on internet today, and this quote is really very meaningful. Since morning I am telling you students, I am just thinking about it, that how much true is this quote. The quote says our greatest weakness lies in giving up. Yes or no? So freely we give up on the things even without thinking. So the quote says our greatest weakness lies in giving up. The most certain way to succeed is why not to try just one more time? Yes or no? This quote I read in the morning and it is having all my heart and mind. Why? Because it is so true. We always give up. We always try to escape with the problems rather than finding their solution. So now here you are going to get solutions to all your problem. Why not to just give one more time to our life, to our dreams, and this time you have to give all your 100%. No student should say, ma'am, I did 99%, I did 98%. No, you all should give this time your 100%, full concentration, full hard work. And let's then wait for the result. So if you really want to get successful, always give yourself one more chance. You never know which chance will be fruitful. So with this quote, I would like to start my today's session on the chapter Anatomy of Flowering Plants. Now, this chapter is just the continuation of the last chapter which we did. The last chapter was all about morphology, where you have studied the external features in the plant. In this chapter, we are going to study about all the internal organization in the plant body. So what is anatomy? Anatomy means dealing with the internal structures. And now in this chapter, we are going to study about that how plants are internally prepared to function. So introduction mein what are we going to write? Here you are going to mention that what do you mean by anatomy? So anatomy is the study of internal system. Anatomy is the study of internal body structures, right? And in this chapter we are going to focus on the tissues which are responsible for making the entire plant body. So that means we are going to study about tissue. The study of tissue is known as histology. So right now we all are going to become histologists because we are going to deal in with tissues. Tissues. What are these tissues? So tissues represent group of cells which join together to perform some specific function. Correct? These cells, they may be similar in origin or they may be dissimilar in origin. So how will you define tissues? First of all, tissue is the second grade of body organization. The first grade was cellular. Now different cells, they join together to perform some specific function. So tissue represent group of cells having a common origin and usually performing a common function. Correct? These cells can be of similar origin. They may also be of dissimilar in origin. Correct? Now as I said, the basic unit of body is cell. The organisms belonging to kingdom Monera and Protista, their body is just represented by single cell. Therefore, the body organization in such unicellular organism is only cellular. Above cellular, the next grade is tissue grade. So how tissues are formed? Different type of cells or similar type of cells, they join together, they interact with each other to perform some specific function, and that structure is known as tissue. So different cells, they join together, or similar cells, they join together to perform a specific function and form a new structure called tissue. Maybe xylem is one of the tissue. Correct? Now, this xylem, which is a tissue, it is made up of some cells. Now, my dear students, this xylem is capable of performing function like conduction. Xylem is responsible for conducting water and mineral from the roots to the leaves. Yes or no? Xylem is made up of so many cells. Now, actually, the function, the property of xylem is due to its cellular interaction. Yes or no? Can you expect that the cell which is actually making the xylem, this individual cell, if I take out this cell which is responsible for making xylem, can you expect that this single cell will also show the property of conduction? No. The individual cell which is making the tissue does not exhibit the property of tissue. The cell is responsible for making the tissue, but independently, individually, this cell will not be able to perform the function what tissue is performing. So, as tissue is a next grade of body organization which is represented by a group of similar or dissimilar cells, thus the property which is exhibited by tissue is the result of cellular interactions. So many cells are interacting. That is why they are able to conduct water and mineral in case of xylem, right? But if you remove one cell and ask this cell to perform the same function in the same manner as xylem does, then this cell is not capable of doing that, because the property of tissues lies in its constituent due to the interaction of its constituent cells, but not in its individual cell. Properties of tissues are not present in their constituent cell. Rather, it arises due to their interaction. Do you agree with this? Yes or no? Correct? Very good. Study of tissue, I have already told you, is histology. Moving ahead. Now, types of tissues. Tissues on their capability. Tissues are made up of cells. Correct? Now, these cells on the capacity of able to divide or not able to divide, tissues can be grouped into two broad categories as meristematic tissues and permanent tissues. Whole chapter is initially based on understanding what are meristematic tissues, what are their functions, what is permanent tissue, and what are their functions. So, important segment of the chapter is now going to start, which is classification of tissues. Tissues on their ability to divide or not can be broadly divided into two groups as meristematic tissues and permanent tissues. So I am going to teach you both the tissues first in a comparative mode and then individually. Correct? So first, what do you mean by meristematic tissues? Meristematic tissues, my dear students, represent group of cells, group of young and immature cells who are capable of dividing. The group of actively dividing cells which are young, which are immature, living cells, correct? And their only job is to divide, to increase the number of cells in the body so that the body can grow, right? Apart from this, meristematic tissues have no other role. They cannot perform any other function apart from division. So what do you mean by meristematic tissue? Meristematic tissue represents group of young, immature cells that are actively dividing, right? Responsible for the growth of the plant body. Responsible for the growth of the plant body. They are made up of usually living cells. Correct? Rich in protoplasm, thin cellulosic cell wall. Simply they are made up of young and immature plant cells. So these are meristematic tissues. Contrary to meristematic, when these group of cells themselves, when they lose their capacity to divide, when they escape the cell cycle, they are not further dividing, they have become mature and now they are ready to perform function like maybe storage, photosynthesis, secretion, excretion, etc. Then those group of cells from meristematic, they become permanent. You can simply say that meristematic give rise to permanent tissues. Meristematic tissues comprises of immature young cell. Now when these immature young cell, when they become mature, they tend to lose their capacity of division. They are now ready to perform some specific role. Maybe storage, maybe photosynthesis, maybe respiration, any function. Correct? So what do you mean by permanent tissues? Permanent tissues represent group of mature cells which have lost their capacity of division. Which have lost their capacity of division, rather they have become ready to perform specific function. Correct? Yes or no? So this is the basic difference between meristematic and permanent. We have more differences that we are going to do as we proceed in the chapter. Now my dear students, on the basis of origin, meristematic tissues and permanent tissues can be further classified. So first, I am going to explain you the types of meristematic and permanent tissues together with the help of some new terminologies and then separately. Now my dear students, initially you know there is a seed. In morphology, I hope you all have seen that video, the last video which we uploaded on morphology. The introduction part, I told you there is a seed. The seed which encloses the embryo. Embryo consists of actively dividing cells. When these actively dividing cells, when they get the favorable condition, they start dividing to produce plumule and radicle along with the cotyledons. Plumule further grow to develop into shoot system. Radicle further grow to develop into root system. Correct? Now, those group of actively dividing cells which are actually present in the embryo. Suppose this is the seed and this is the embryo. Now, within this embryo, those group of actively dividing cells are known as promeristem. Normally actively dividing cells, meristem. But promeristem means the group of actively dividing cells found in embryo. So embryo consists of promeristem cells. Number 1 point. These promeristem cells, when they get the favorable condition, they start dividing to give rise to plumule and radicle. So now when this embryo will get the favorable condition, what will happen? Embryo will start germinating into plumule and radicle. Now, the tip of plumule is having some actively dividing cells and the tip of radicle is also having some actively dividing cells. Now, these actively dividing cells are known as meristem. Which meristem? Primary meristem. Primary meristem represents those group of actively dividing cells which directly arise from the embryo of the seed. Now, these meristem are actually responsible for primary growth of the plant body. Means, now this tip of the plumule, actively dividing cells. Tip of the radicle, actively dividing cells. They are going to divide and to produce a complete new plant with well-developed root system and well-developed shoot system. So this is called as primary body of the plant, right? So first you have promeristem. Promeristem give rise to primary meristem. So how will you define primary meristem? Primary meristem represents those group of actively dividing cells which arise directly from the promeristem. They are the first type of cells that are responsible for growing the plant body. Now, primary meristem cells, they are dividing. Now, parallelly when the cells are dividing, they are also getting mature to perform function so that they can either produce roots, stem, leaves, flowers, etc. Yes or no? So the cells of leaves, the cell of stem, the cell of flower, they are mature cells. They are performing function like either photosynthesis, conduction, etc. So those cells are actually arisen from primary meristem. When primary meristem, the cells of primary meristem, when they initially lose their capacity of division to perform some specific function, then those group of cells, those tissues are known as primary permanent tissue. And formation of primary permanent tissue from primary meristem is known as differentiation. What is differentiation? Differentiation is the process by which primary meristematic cells, when they lose their capacity of division, when they become ready to perform function, to produce primary permanent tissues. So this process of conversion of PM into PT, primary permanent tissues, PMT, primary meristematic tissue, converting into primary permanent tissue. So this conversion is known as differentiation, right? Now, so primary meristematic tissue and primary permanent tissue together, they make the primary plant body, the basic plant body which you see. Now, in some plants like specifically dicots, you see that with time, with years as they pass on, their stem and their root, they become thicker, right? In first year they were very slim and thin, but second year they become slightly more thicker, third year more thicker. So how is that happening? What is that phenomena? That phenomena is called as secondary growth, correct? And how the secondary growth takes place? Now, the primary permanent tissues, there are some primary permanent tissues which temporarily lose their capacity of division. Now, when these primary permanent tissues, if they regain their capacity of division, then they will form secondary meristematic tissues. Yes or no? And conversion of primary permanent tissue into secondary meristem is known as dedifferentiation. So what is dedifferentiation? Formation of secondary meristematic tissue from primary permanent tissue. Right? Now, secondary meristematic tissue, like for example lateral meristem, I'll tell you. Now, these lateral meristem, they appear later in the plant body. Since beginning they are not present. They come in the plant body once primary growth is done. And formation of lateral meristem is not a feature of all plants. They are observed in dicots. So when lateral meristem, they appear in the plant, now what will happen? Again, they are group of actively dividing cells. The cells will divide, they will increase the number, and a time will come when those cells will become mature, will again lose their capacity of division to form secondary permanent tissue by the process of redifferentiation. Correct? So, there are 3 steps: differentiation, dedifferentiation, redifferentiation. Differentiation is actually formation of primary permanent tissues from primary meristematic tissues. Primary meristematic tissues, like for example apical and intercalary. Apical and intercalary. Correct? Now these apical or intercalary meristem are responsible for just increasing the plant height. Now once the cells of apical and intercalary meristem, they mature, they lose their capacity of division, now they are ready to perform function, then they form primary permanent tissues like parenchyma, collenchyma, sclerenchyma, xylem, phloem, etc. Correct? Now, this conversion of primary meristem into primary permanent is differentiation. This differentiation is responsible for the formation of basic plant body, the primary plant body. But in some plants like dicots, they need some secondary growth, they need some extra growth to increase the thickness of their stem and root. For that, some primary permanent tissue, specifically parenchyma, they regain their capacity of division which they have lost initially. They regain their capacity of division by the process of dedifferentiation and get converted into secondary meristematic tissues. Secondary meristematic like lateral meristem. So lateral meristem has actually arisen from primary permanent. And lateral occurrence of lateral meristem is not since the beginning. This basic plant body which I have drawn over here, it is not having lateral meristem right now. But later, if it is required, then by the process of dedifferentiation, secondary meristem, lateral meristem will appear. Now when lateral meristem will come, now what will happen? They are a group of actively dividing cells. The cells will divide, will increase the number, and a time will come when again they will lose their capacity of division. That next time when they lose their capacity of division is known as redifferentiation. Re means again. First time differentiation is done, primary permanent is produced. Now again the meristematic tissue are undergoing differentiation. So redifferentiation to form secondary permanent tissues like secondary xylem, secondary phloem. Yes or no? And this process of redifferentiation is responsible for secondary growth. The process of differentiation is responsible for primary growth of plant body, which you see in all plants without any exception. But presence of secondary growth is restricted to some plants like in dicots. Correct? Yes or no? So how many steps are there in overall growth of the plant body? 3 steps. Differentiation, dedifferentiation and redifferentiation. First thing, on the basis of origin, how many meristems are there? 2. Primary meristem, secondary meristem. On the basis of origin, how many permanent tissues are there? Primary permanent, secondary permanent. So I hope all the types of meristem as well as permanent tissues on the basis of origin is clear. Now we are heading towards the first type of primary meristematic tissues. We are going to study them on the basis of location. So primary meristematic tissues are those group of actively dividing cells which directly arise from the promeristem. First thing. Second, on the basis of location, they can be of 2 types. That is apical meristem and intercalary meristem. These 2 are the group of actively dividing cells which are present initially in the plant body. Their function is to help in the increase in the length of the plant. And now let's see where are they located. So if I first talk about apical meristem, then apical meristem, apical itself means tip. So these are those group of actively dividing cells which are present at the tip of the root or shoot. Correct? Now these are group of actively dividing cells present at the tip of the root or shoot. Right? Now, if they are present at the tip of the root, then it is known as root apical meristem. And if it is present at the tip of the shoot, then it is known as shoot apical meristem. The main role is divide, increase the number of cells and allow increase in the length of the plant body. If I specifically talk about shoot apical meristem, the diagram of shoot apical meristem is there in your syllabus. Let's try to discuss it. So here I am just going to draw a simple diagram of shoot apical meristem with some labelings. Now, please don't laugh on this. See, these are the leaves. Correct? This region is actually the node from where the leaves are arising. Now initially this is the tip of the shoot which is known as shoot apical meristem. Shoot apical meristem consists of group of actively dividing cells that is known as zone of meristem. Now what happens, students, when this, now here right now is the tip of the shoot. Here are present group of actively dividing cells. A time will come, new cells will be deposited and the length is going to increase further. So when the length is going to increase further, initially SAM is over here. But as the length will increase, this SAM will shift to the next region. Correct? From every SAM, the leaves will arise. So you can consider that initially this shoot apical meristem, the zone of meristem would have been present over here. Now, when the cells divided, new cells deposited, length increased, so the shoot apical meristem shifted to the next region, increasing the length, marking the increase in the length. Right? Now, what happens while the shoot apical meristem is growing upward, some of the actively dividing cells are left behind in the axial angle of the shoot and the leaf. Here, some group of actively dividing cells are left behind that constitute axillary bud. You remember in morphology I taught you there are two types of buds, there are two types of compact shoot system, axillary and apical. Apical bud is actually this, the tip of the shoot where actively dividing cells are present. Their role is to increase the plant in length, in height, like this. Whereas the axillary buds which are present in the axile angle of the leaf and the stem is responsible for increasing the plant body laterally by producing either a flower or a branch. Now how this axillary bud arises? Now you see. These axillary bud arises when group of actively dividing cells called shoot apical meristem while growing upward leave some of their cells behind in the axile angle of the leaf and the shoot. So this group of actively dividing cells make bud. Which bud? Axillary bud. What is the role? To give rise to either a new branch or a new flower. Correct? So this much you have to do for apical meristem. First point, where are they located? Second point, what is their function? Third point, how axillary bud has come into existence. Now moving towards the second type of primary meristematic tissue, that is intercalary meristem. Now if I talk about intercalary meristem, the name itself will explain you. Inter means in between. Intercalary means in between. Now what in between? These are those group of actively dividing cells which are present in between two mature tissues. So what is intercalary meristem? These are those group of actively dividing cells which are present in between two mature tissues. Right? Now what is this? What do you mean by this, that they are present in between two tissues? Now what will be the location? So now see, you know the concept of nodes and internodes. Nodes represent actively dividing regions on the shoot, and the gap between two nodes is internode, and internode consists of mature cells, those cells which have lost their capacity of division. So suppose this is my stem, correct? On the tip of the stem will be present shoot apical meristem. While the shoot apical meristem is growing, nodes are produced from where the leaves are arising. So these are the nodes, and the gap between two nodes is known as internode. This is node, this is node. But the gap between two nodes that consist of what? Mature tissues, the cells which have lost their capacity of division, makes internode. Now, so this is one internode, this is second internode. Intercalary means those group of actively dividing cells which are present in between two mature tissues. So see, this is one mature tissue, this is second mature region. Now these group of actively dividing cells are actually present in between them, near node here. Correct? So the blue one which I am marking is actually intercalary meristem. Intercalary meristem are present in between two mature tissues just above or below the node. Now what is their role? The cells over here are going to divide. When they will divide, they will allow to increase the length of the internode. The gap between two nodes will increase, thus elongating the stem. Correct? So their role is the increasing the length of the internode, because here division will take place, new cells will deposit, here new cells will deposit, new cells. So what will happen? The internode length will increase. Now if internode lengths are increasing, definitely overall the shoot length is also going to increase. So what is the role of intercalary meristem? Increasing the internode length. Full stop. Thus increasing the shoot length. They are directly proportional. Do you agree with this or not? Very good. Apart from this, intercalary meristem, my dear students, in grasses specifically, they are used for regenerating the lost grass due to herbivory, due to grazing. Now what happens? See, in last lecture only we have studied about grasses. They vegetatively propagate by runner method. So suppose this is the soil, right? Here I have planted my grass. So from a node that is present near the ground, a short lateral branch will arise, and then when this branch will touch the soil, it will again produce a new grass. Again from the node which is present below the soil, what will happen? A branch will arise horizontal to the soil. Again it will get the favorable condition, touch the soil and produce a new grass. So isn't it like this grasses propagate, and this method of propagation is runner, a subaerial stem modification. Correct? Now, I also told you one thing, that these branches can be called as internode. Why? Because here is a node, here is a node, and this branch is actually made up of mature cells, and they are present between two nodes. Therefore, they can be called as internode. So actually this branch can be called as internode. This is also internode. Now what is intercalary meristem? Where are they present? They are present in between two internodes, above or below the node. So that means here can be your intercalary, here intercalary, here can be intercalary, intercalary meristem. Can you see the white regions? Correct. Now what happens during grazing? When an animal will come, that animal is going to remove the grass, will eat the fleshy green leaves of the grass. Now these intercalary meristem which are present near to it, they are going to divide, produce new cells which will mature to form leaves. Thus, intercalary meristem in grasses are also responsible for the regeneration of leaves, ones which are lost due to grazing. Clear? Can you see with the help of the diagram that where the intercalary meristem are located, and it is so easy for these meristems to produce new grasses? Correct? So what you can write, that intercalary meristem helps in regeneration of grasses which are lost due to herbivory. So this is about apical meristem and intercalary meristem, which are the two types of primary meristematic tissue on the basis of location. Moving ahead with secondary meristematic tissue, types of secondary meristematic tissue. First you should know what is secondary meristem. Already know. The group of actively dividing cells which arises due to the dedifferentiation of primary permanent tissue as and when required. These are actively dividing cells which appear later in the plant body. They are not present since the beginning. And their role of such secondary meristematic tissue is to help in secondary growth, that is increasing the thickness of the root and shoot. Is it observed in all plants? No, it is only observed in some specific group of plants like in dicots. Clear? Shall I write all these points? Come on. So secondary meristem means group of cells. Group of which type of cells? Group of actively dividing cells. that arises due to which activity? De-differentiation. You know it. That arises due to de-differentiation. Right? So they arises due to de-differentiation of primary permanent cells. First point. Second point. What will be the second point? They are not present since the beginning. These meristem, they are not present since the beginning. They appear later in the plant body and are responsible for secondary growth. They are responsible for secondary growth. Right? One example in secondary meristem is lateral meristem. So lateral meristem is also known as secondary meristem. ठीक? Now, what do you mean by secondary growth? Secondary growth means increasing. Secondary growth. See, in primary growth, there is only increase in the height. Now, height is increased. The primary growth is done. Jitna height ko increase hona tha, the increase is done. Now, in secondary growth, thickness is going to increase. The plant is going to increase in the diameter. So, secondary growth means increasing the diameter, the thickness of plant body. That is root and shoot. Only. Secondary growth is absent in leaves, flowers, fruits. No. In them, no secondary growth takes place. Secondary growth is only taking place in the root and shoot. And that too also not in all plants, in only some plants like dicots. Now, why only in root and shoot secondary growth takes place? Because lateral meristem, which is responsible for secondary growth, is only found in root and shoot. Where are they located? See now. Now, if this is the plant body, right? Having distinct nodes and internodes, this is the stem I am drawing. Having distinct nodes. So, these are the nodes. The gap between two nodes is internode. Right? So, this is the tip region. This is the tip of the shoot. So, tip of the shoot will have shoot apical meristem. The gap between two nodes, internode. In between two internodes, above and below node, will be present your intercalary meristem. So, this region is rich in intercalary meristem. Intercalary meristem. These two are the type of primary meristem. Now, if I talk about secondary meristem, then they develop on the lateral sides of the plant body, both in the root and shoot. Here, they develop on the lateral side of the plant body. That is why they are known as lateral meristem. Yes or no? Why are they known as lateral meristem? Because they develop on the lateral side of the plant body. What they do? They are going to increase the thickness of the root and shoot. They are going to increase the plant body in its diameter, in its girth. When they occur, see, SAM and ICM, they are present since the beginning. Their work is to increase the height of the plant body. But secondary meristem comes later on. And what they do? They increase the thickness. Am I clear? Yes or no? Lateral meristem are also known as cylindrical meristem. The other name for lateral meristem is cylindrical meristem. Clear? Now, this is about the location and function of lateral meristem. Now you will see the examples of lateral or secondary meristem. So the three examples of lateral meristem, rather I should say two examples of lateral meristem are Vascular Cambium and Cork Cambium. So the two examples are Vascular Cambium, I'll tell you what are these, and Cork Cambium. Vascular Cambium, my dear students, are of two types, that is intravascular cambium and intervascular cambium. Intra and intervascular cambium. So overall there are three examples of lateral meristem: intravascular, intervascular, together called as vascular cambium, and next is Cork cambium. Vascular beta means xylem and phloem together. So the actively dividing cells which develops in between vascular bundles are known as vascular cambium, which can be of two types, intra and inter. Now let me tell you in details about what is intra, what is inter, and what is Cork cambium. So now, okay, let's move ahead. Since I don't have some extra sheet attached to it, right. So let me use this page only. This question we will solve later, but I'm going to use this page initially to explain you the examples of lateral meristem. Right? So how many examples of lateral meristem I have told you? Cork cambium and vascular cambium. First, let's discuss about vascular cambium. Vascular means what, beta? Vascular means xylem and phloem. Bundles of xylem and phloem. Xylem and phloem, they are primary permanent tissues which are already present in the plant body. Correct? Now, for example, I am going to explain you this with the help of vascular bundles present in dicot stem. In case of dicot stems, vascular bundles are arranged in ring-like manner. Just please focus. This is a cross section, this is a circular section of a dicot stem. In center of the stem is pith. Now, around this pith are present bundles of xylem and phloem. Correct? Xylem on the inner side and phloem on the outer side. Just try to understand. Right? Now, within each bundle, in case of dicot stem, already some group of actively dividing cells are present. Within each bundle, already some group of actively dividing cells are present, called as intravascular cambium. Intra means within, fascicular means bundle, cambium means group of actively dividing cells. So intravascular cambium is present already within the bundles of xylem and phloem. My dear students, this xylem and phloem, they are primary in nature. That means they are found since the beginning. Since the primary growth of plant body was taking place, then only primary xylem and primary phloem was formed. Now, if primary xylem and primary phloem are present since the beginning, then surely intravascular cambium will also be formed since the beginning only. Yes. So intravascular cambium is a lateral meristem, but primary in origin. It is an exception. It is lateral meristem. It is going to help in secondary growth, but because it is formed earlier, it does not appear later in the plant body. It is present since the beginning. That is why it is an exception, and it is primary in origin even after being lateral meristem. Even after being lateral meristem, intravascular cambium is primary in origin. Right? Now, in between each bundles, there are some parenchymatous cells present, which are again primary permanent tissues, right? Now, when the right time comes, these primary permanent tissues, they lose, no, they gain. They regain their capacity of division to form lateral meristem, interfascicular cambium. A right time will come when these parenchymatous cells that are present in between vascular bundles, they will regain their capacity of division to form what? To form interfascicular bundle. Why inter? Because these are present in between two. Fascicular means bundles. Cambium means group of actively dividing cells. So intrafascicular cambium is present since the beginning. Interfascicular cambium appears later as when required for secondary growth. So these two both, intra and inter, both help in secondary growth. They both are the types of lateral meristem. They both are the types of vascular cambium. One present within the bundle, the another present between two bundle. The one which is present within the vascular bundle, that vascular cambium is primary in origin, called intrafascicular cambium, and another lateral meristem, another type of vascular cambium which is present between two vascular bundle, is interfascicular, and it appears later in the plant as and when required. Correct? So these are the two types of examples of lateral meristem. Which lateral meristem? Vascular cambium. Apart from this, there is one more type of lateral meristem that appear called as Cork Cambium. And this Cork Cambium I will explain you once we will do secondary growth. Yes or no? Let me just give you a quick revision of it. That is, what is type of secondary meristem? So there is one example in secondary meristem, that is lateral meristem, also known as cylindrical meristem. They appear later in the plant body and they are responsible for increasing the thickness of the plant. They are present on the lateral sides of the plant. First thing, they are not found in all plants. They are restricted to some plants like dicots. Example में दो याद रखने हैं, vascular cambium and cork cambium. Vascular cambium can be of two types, intra and interfascicular cambium. Intrafascicular cambium are those group of actively dividing cells which are present within one bundle, and they are primary in origin, which becomes an exception. Moving ahead, if between two vascular bundles some group of actively dividing cells are formed, then they are called as interfascicular cambium. Correct? Yes or no? See this. So one is interfascicular, another is intrafascicular. Together they are known as vascular cambium. And this is an example of lateral meristem. Apart from vascular cambium, there is one more example called cork cambium. These two together, they help in secondary growth. This is you have to study for lateral meristem, and this is all about primary meristematic tissues. What you have done for primary meristematic? First, what is meristem. What do you understand by the term meristem? Second, types of meristem on the basis of origin, primary and secondary. Third, types of primary meristem, that is apical or intercalary. Types of secondary meristem, that is lateral meristem. This much you have discussed. And on this basis, I have one question for you. The question says, lateral meristem are secondary in origin except. Now, the answer is also on the same page. Question is also on the same page. Options are vascular cambium, cork, intravascular, none of the above. So, vascular cambium includes intra and inter both. However, intervascular cambium is secondary in origin. It is not an exception. So, vascular cambium becomes wrong. Cork cambium, purely secondary in origin, thus not the answer. Intravascular cambium, see, intravascular cambium is primary in origin. It is present since the beginning. It is present even in the primary plant body. However, it is functional later on. Thus, this becomes an exception as it is a lateral meristem but not primary, but not secondary, rather it is primary. So, the answer of this question becomes C. Clear? I hope meristematic tissues is clear to each and every student. Now, my dear students, it's the time to start with permanent tissues. The question I hope is clear to everybody, which we did for lateral meristem. Now, with this, your meristematic tissue is over. Moving ahead in the chapter, next group of tissues which have lost their capacity of division are permanent tissues. Now we are going to study about the next group of tissues called as permanent tissues. Right? Now, permanent tissues can be divided into two broad categories. First, simple permanent tissues and complex permanent tissues. Simple permanent tissues are those group of cells which are made up of one type. So, group of similar cells which are ready to perform function. So, group of similar cells which are ready to perform function are simple permanent tissue. On the other hand, complex permanent tissues are those which represent group of dissimilar types of cell. When one or more type of cells they join together to perform a specific function is known as complex permanent tissue. So these represent group of dissimilar cells. Right? In simple permanent tissue, you have to study three types: parenchyma, collenchyma and sclerenchyma. Whereas in complex permanent tissue, we are going to study about xylem and phloem. So one by one, we are going to see all the types of simple as well as complex permanent tissues. First, let's start with simple permanent tissue, and that too parenchyma. So first type of simple permanent tissue which is found in majority of the plant body. If I talk about major portions of the plant body, then it is made up of parenchyma, which is one of the simple permanent tissue. So first point you have to remember for parenchyma is it represents the major portion of the plant body. Majority of organs in plant are made up of parenchyma. Right? Second point related to parenchyma. The cells of parenchyma, if I talk about, they are isodiametric. See, diagrammatically also parallelly I will show you. Cells, all the parenchyma cells, they are almost of similar in size. So when all the cells are similar in size, then we use the term isodiametric. Right? Here in the diagram you can see that the space between cells is very less. Correct? So third point comes that intercellular spaces in case of parenchymatous cells is less or either absent. So third point you can write is that intercellular spaces are minimum. Correct? Fourth point related to parenchyma, which is very important to learn, is about the nature of the cell. The cells, they are young cells, they have thin cellulosic cell wall. Correct? So these parenchymas are the plant cells which possess thin cellulosic cell wall, and they are living as they are rich in protoplasm. So what about their cell wall? Parenchyma, they possess thin cellulosic cell wall. These cells are living in nature. Why? Because they are rich in protoplasm. Central, there's a large vacuole, and the protoplasm, the cytoplasm, is shifted towards the periphery. Right? So these are certain points related to parenchyma that you have to remember. Along with this, let me write one important more thing, that is the function of parenchyma. Wherever in plant body parenchyma is present, they have some specific functions like either storage. So number one duty of parenchyma in plant is storage. Second important duty is secretion. Secretion of the material that are prepared by the plant itself. Now, if some parenchyma, they may also develop chloroplast. Now, if in any part of the plant body that is made up of parenchyma, and if their parenchyma contains chloroplast, then that region can also perform photosynthesis. Like if I talk about mesophyll cells of the leaf. Mesophyll cells of the leaf are parenchymatous in nature, and those parenchyma cells, they have chloroplast. Hence, mesophyll cells in the leaf can perform photosynthesis. Correct? So, parenchyma containing chloroplast can perform photosynthesis. Apart from this, parenchyma also helps in storage and secretion. So, these are some major functions that are performed by parenchyma. Moving ahead with the second type of simple permanent tissue is collenchyma. Collenchyma are also group of simple permanent tissues which are majorly found in dicot stem. If you cut the section of a dicot stem, then the topmost layer is epidermis. Below epidermis comes hypodermis. So, in case of dicot stem, the hypodermis of dicot stem is collenchymatous. Right? It is collenchymatous. Usually presence of collenchyma is absent in monocots. Monocots usually they do not have collenchyma as simple permanent tissue. But in dicot stem, you will find the presence of collenchyma in their hypodermis. First thing. Second thing, their cell wall is also made up of cellulose. Apart from cellulose, they contain hemicellulose and pectin as well. Collenchyma are also living cells, also provide mechanical strength. So second point which you have to remember for them are that collenchymatous cells, they are living in nature. Cell wall is made up of cellulose. Along with cellulose, they also have the deposition of hemicellulose and pectin. Correct? Now, let's see the diagram of collenchyma as well. Suppose these are collenchymatous cells. They may not be isodiametric, right? Now, what happens in case of collenchyma? The intercellular spaces which you see right now is actually completely absent. Why? Why intercellular spaces are completely absent in case of collenchyma? Because the space, the corner of each cell, in case of collenchyma, they are thickened due to the deposition of cellulose, hemicellulose and lignin. So corners of each cell, they get thickened due to the deposition of such chemicals. As a result, in case of collenchyma, intercellular spaces are completely absent. Whereas in case of parenchyma, you saw that intercellular spaces may be present. However, in case of collenchyma, these spaces are absent as corners are thickened due to the deposition of chemicals like cellulose, hemicellulose and pectin. Right? Clear to everyone? Yes or no? Very good. Now moving ahead with the functions of collenchyma. Collenchyma beta, they may also possess chloroplast. If they possess chloroplast, then collenchyma can also help in photosynthesis. Right? Apart from this, the main function of collenchyma is to provide mechanical support to the growing part of the plant body. Mechanical support to the growing parts of plant body. Like for example, if I talk about tip of the petiole. Suppose this is a stem. Here is the petiole. This is a stalk of the leaf on top of which the leaf is present. So this is the tip of the petiole. This tip of the petiole will have collenchyma. Why? Because the young leaf needs support. And collenchyma at the tip of the petiole will provide that support. Clear to everybody that what are collenchyma? Where are they found? They are absent in case of monocots. Now, moving ahead with the third type of simple permanent tissues, that is sclerenchyma. If I talk about sclerenchyma, then they are different from parenchyma and collenchyma. How? Parenchyma and collenchyma are the two living types of simple permanent tissue. Their cell wall is rich in cellulose. They have a living protoplasm. But if I talk about sclerenchyma, then sclerenchyma are dead cells. They do not have, towards maturity, the cells of sclerenchyma, they lose their protoplasm. Their cell wall starts depositing lignin, which is a dead material. Now they are dead, so they cannot perform any functions like photosynthesis or such kinds of function. So the main role of sclerenchyma in that case becomes to provide mechanical support. Wherever sclerenchyma will be found in the plant body, there its role will be to provide strength, rigidity and support. Clear? Shall we write these points for sclerenchyma? Yes, ma'am. Very good. So what are you going to write for sclerenchyma? Number 1 point, these are dead cells. Why are they dead cells? Because they are devoid of protoplasm. So when sclerenchymatous cells on maturity, they lose their protoplasm and thus they become dead. Apart from this, their cell wall is rich in a substance called lignin. And this lignin is also a dead material. Right? Correct, everybody? What is the main role? The main role is to provide mechanical strength. The main role is to ensure mechanical strength and support to the plant. Now, sclerenchyma on the basis of their shape, development and origin can be of two types. They can be of how many types? They can be of two types. So, sclerenchyma on the basis of shape, origin or development, they can be of two types. Sclerenchymatous fibers or sclerenchymatous sclerides. First, I'll talk about fibers. Sclerenchymatous fibers, my dear students, they are long. Sclerenchyma, when they are present in the form of long pipe-like structures with tapering ends. So, fibers are long cells with tapering ends, spindle-like body. So, sclerenchyma which are present in the form of fibers have spindle-shaped body. Correct? This is how sclerenchymatous fibers will appear to be. Now, they do not have protoplasm in the center. So, the center space will be called as lumen. Their cell wall is highly thick. Why? Due to the deposition of lignin. So, their cell wall is rich in lignin. More the lignin deposition, the lumen will become narrow. Obviously, you deposit more and more lignin, the space within the cell will become narrow. Correct? So, this is how fibers appears to be. They don't have any cell organelles. The space is devoid of protoplasm and that empty space is known as lumen. Now, therefore, fibers, they act like hollow pipes. Correct? They act like hollow pipes. Hence, they can help in conduction of water and mineral apart from providing support. Similarly, coming to sclerites. Sclerites are spherical shaped. They are spherical shaped sclerenchyma which are richly found in the fruit pulp of guava, chiku. I don't know if you have eaten chiku, must have really eaten chiku. When you eat that chiku, some gritty kind of texture you feel in your mouth. When you eat guava, some gritty kind of texture you feel in your mouth. Why that texture is not when you eat banana or when you eat mango? Because in the pulp of guava and chiku, sclerides are present and these sclerides give that gritty texture to the fruit which is absent in case of banana or mango. Apart from the fruit pulp, these sclerides can be found in the leaves of tea, right? They are also found in some fruit walls of some famous nuts. So sclerides, they are found in different parts of the plant body. Some examples I have told you. They are spherical shaped sclerenchyma. Their main function as being sclerenchyma will be to provide mechanical support and strength. Now, similar grounds like fibers, they also have lumen deposition, right? In the center they have lumen, but the lumen is very, very narrow. In fibers still lumen space is there. But when we come to sclerites, lumen becomes highly narrow due to heavy deposition of lignin. Overall, everything is lignin. So lignin is so much deposited in case of sclerites that the lumen becomes very narrow. Now, how conduction will take place? Lignin is impermeable. So how conduction will take place? Hence, this lumen is or this lignin is obliterated. Some pits are formed like this. Let me use white color. Hope it is going to function. Yeah. So like this, some of that's why we say that the lumen is obliterated in case of sclerides. Why obliterated? These pits, why are they produced? Because there is so much lignin deposited that the lumen becomes very narrow. And thus, to allow conduction, some openings should be made that joins the lumen to the outside region of the cell. Therefore, some passages are made which is known as obliterated lumen. Correct? So this much you have to understand and learn about simple permanent tissue. Parenchyma, collenchyma and sclerenchyma. Hope, my dear students, all the three types of simple permanent tissues are clear. Now we are moving ahead towards complex permanent tissues. The complex permanent tissues are of two types, xylem and phloem. What is complex permanent tissues? Complex permanent tissues represent group of dissimilar cells that joins together to perform some specific function. First, we are going to study about xylem. Right? Xylem is not a new term. You all have heard when you must be in class 9th or 8th, right? What is xylem? Now I ask you a question. What is xylem? How will you reply? What will be the first or the opening statement? Your first and opening statement should be that xylem are the conducting tissues found in the plant body that are responsible for the movement of water and mineral. Where to where? From roots to the leaves. They are unidirectional. Always xylem is going to function from root to leaf. Never ever xylem will function from leaf to root. So you can say that xylem are conducting tissues. Number 1 point. What do they conduct? They conduct water and mineral. Where to where? Always from roots to leaves. Because their direction is fixed, hence we can say that xylem are unidirectional. Clear? All the 4 points? Very good. 5th point. Xylem, my dear students, they are rich in sclerenchyma. They are rich, they are made up of fibers. So we can say that xylem are the dead complex tissue. Overall, if you see, then xylem, they are made up of dead cells. Hence, xylem as a conducting tissue is considered as dead in nature. Now, xylem is a collective term for 4 different components. We can say that xylem is made up of 4 different components. Xylem tracheids, xylem vessel, xylem fiber and xylem parenchyma. The first 3 which I said are sclerenchymatous. But the 4th one which I said, as the name suggests, xylem parenchyma, they are the exception to xylem. They are made up of parenchyma, and when they are made up of parenchyma, that means they are living in nature. Clear? Chalo. So let's start with xylem. First point, xylem is a complex permanent tissue that functions as a conducting tissue responsible for the movement of water and mineral from roots to stems and leaves. They are unidirectional. Very, very important. Right? Apart from this, it also provide, definitely they are dead in nature, so they can also provide mechanical support and strength to the body. Xylem is made up of 4 different elements, that is tracheids, vessels, fibers and parenchyma. The first 3 are dead components, but the 4th one is the living component and it becomes an exception. Clear? Very good. Moving ahead. Now, next point which you have to remember for xylem is formation of such conducting tissues, that is xylem and phloem. It begins from pteridophytes. You have already studied plant kingdom. If you have not studied, then please go through the last video, last video which I released on plant kingdom. So my dear students, in plant kingdom, from pteridophytes onward, the formation of conducting tissues begin. So pteridophytes, gymnosperms and angiosperms, all 3 have conducting tissues. xylem and phloem. However, if I talk about pteridophytes and gymnosperms, they have xylem, but in their xylem, vessel is not present. Formation of vessel starts from angiosperms. So, presence of vessel is a characteristic feature of angiosperms. If I talk about gymno and pterido, they have xylem, no doubt. But in their xylem, tracheids are the major conducting tissues as they lack vessels. But when it comes to angiosperms, in angiosperms, all the four conducting tissues in the xylem are present, and in angiosperms, the chief conducting tissue for water and mineral becomes vessel more than tracheids. Clear to everybody? Okay, moving ahead. So I hope we should write this point. Gymnosperms lacks vessels in their xylem. Gymnosperms as well as pteridophytes lacks vessels in their xylem. Formation of xylem is a characteristic feature of angiosperms. Sure? Very good. So these are certain points that you have to remember for xylem. Now, you remember meristematic tissues on the basis of origin, primary meristem and secondary meristem. Primary meristem give rise to primary permanent tissue, whereas secondary meristem give rise to secondary meristematic, secondary permanent tissues. So when there are in dicots, when there are two types of permanent tissues, primary and secondary, so that means there will be two types of xylem. So they can be on the basis of origin. There are two types of xylem. That is primary xylem and secondary xylem. Primary xylem arises by the differentiation of primary meristematic tissues, whereas secondary xylem arises due to the redifferentiation of secondary meristematic tissues. Remember the initial part of the lecture? Yes. Now, primary xylem again can be of two types. This is primary xylem and secondary xylem. Primary xylem again can be of two types, that is protoxylem and metaxylem. The initial formed xylem tissues after the differentiation of primary meristematic is known as protoxylem. Protoxylem, comparatively, they have narrower vessels, whereas metaxylem are later formed xylem tissues with wider vessels. So you should know this point, that protoxylem have, first point you should know, that protoxylem are first of all initially formed xylem with narrow vessels. Correct? On the other hand, if I talk about metaxylem, then they are later formed xylem tissues with wider vessels. Now students, can you guess that which type of xylem, proto or meta, will be beneficial for conduction of water and mineral? Any guesses? Protoxylem or metaxylem? Please mention in the comment box. See, of course metaxylem. Why? Because they have wider vessels. The space within the tube, if it is more water will pass. If the space within the tube is less water will pass. So more efficient type of xylem is metaxylem. Why? Because in them they have wider vessels and they are later formed xylem. Clear? Yes or no? So we can say that protoxylem are comparatively little bit less efficient as in the case of meta and proto. Now, depending upon the arrangement of protoxylem and metaxylem, again we can have two types. Depending upon the arrangement of these proto and metaxylem, there are two categories. Endarch, which is seen in case of stems, and exarch, which is seen in case of roots. Now, what is endarch and what is exarch? See, all these vascular bundles, that means xylem as well as phloem, they are present around the pith. Suppose this is a cross section of the stem as well as root. Center is pith, right? We'll deal in with pith in detail as we proceed in the chapter. So this is the pith. So the first type of vascular bundle which is present around pith are usually xylem. So if endarch arrangement is there in case of xylem, then protoxylem, the ones with narrower vessels, will be present close towards the pith, followed by metaxylem. So can you see I am drawing yellow color is metaxylem and green color is protoxylem. So when protoxylem is towards the center followed by metaxylem, then this arrangement is known as endarch. Opposite to this is exarch. What happens in exarch? Just the reverse. When metaxylem, the bigger ones, the ones who have broader vessels, are present towards the pith and followed by protoxylem towards the periphery, then this arrangement is known as exarch. Can you see? The green one, protoxylem, is towards the periphery, and yellow one, that represents metaxylem, are present towards the center. So this type of arrangement is exarch, and when protoxylem in the center followed by metaxylem, then this type of arrangement is known as endarch. Please remember this. Questions on endarch and exarch are usually asked in NEET. They confuse the students. Students get confused. Protoxylem, metaxylem, inner, outer, periphery, center. So please, no need to get confused. Revise it properly. Read the concepts properly with full concentration. Surely you will remember it even in your exams. Right? Now, moving ahead. So now you have seen the basic points of xylem along with their function. Now we are going to talk about all the tissues, all the subcomponents of xylem one by one. First, starting with tracheids. So tracheids are the most common type of xylem tissues which are found in pteridophytes as well as gymnosperms as well as angiosperms. These tracheids, they are represented by long elongated fibers. They are sclerenchymatous. Tracheids are dead in nature. They are made up of sclerenchymatous type of cells. And which sclerenchymatous? Fibers. So tracheids are represented by long elongated fibers whose ends are tapering. So one above the other, cells are kept like this. Tracheids are elongated, tube-like cells with thick and lignified wall with tapering ends. Obviously, they are made up of fibers, so the cell wall is going to be thick. They are dead cells. Why? Because the cell wall shows the deposition of which material? Lignin. Correct? More lignin, less will be lumen. So this is the lumen. And here is the lignin. Right? One elongated cell is kept above the other, then above the other. Like this, they form a whole pipe, and that allows the conduction of water and mineral. Right? These are dead or living cells. Of course, they are dead cells. First point. moving ahead. Some scientists observed when you cut the section of tracheids, then when you see the deposition of lignin, then there are different forms in which lignin are deposited in each cell of tracheid. Their lignin deposition in some may be ladder-like, in some it may be ring-like, in some it may be pitted deposition. So there are different forms of lignin deposition in case of tracheids, right? So first point, I hope it is clear. Second, they are dead cells, you know, they are devoid of protoplasm. Very well you know it. The inner layers of the cell wall have thickening of lignin, and lignin deposition thickening varies in shapes. Some may be scalariform, pitted, ladder-like, reticulate, etc. Spiral thickening can also be there like this. So lignin may be deposited in the form of spirals in their cell wall, right? Now, what is their role? Their role is transportation, and presence of tracheid is found in all tracheophytes of plant kingdom, right? I hope tracheid is clear. Now, moving ahead towards the second component of xylem, which is vessel. Vessels are also known as trachea. Very important. The other name of vessel is trachea. Tracheid plus trachea, together it is known as stretchery elements. And in angiosperms, these stretchery elements help in the conduction of water and minerals. Clear? Thumbs up? Right. Moving ahead. Now, vessels also known as trachea. Vessels, my dear student, they appear to be longer than tracheids. If I talk about whole pipe of vessel, then they appear to be longer than tracheids. However, if in depth I go, then they are actually shorter. When with respect to one cell, if I talk about, see, in case of tracheids, one cell represent one tube, right? One cell represents one tube. Because the cells are long, elongated, this is actually one cell, it is one tube. This is another tube. This is another tube. So in case of tracheids, because the cells are long, elongated, each cell represents one tube. However, in vessels, it is not so. In vessels, many cells, each cell called as vessel member, they are dumbbell-shaped cells, placed one above the other. The common wall between them dissolve and forms a long pipe. So that one long tube is actually made up of many cells, right? So in case of vessel, you should remember many cells make one tube. So surely vessels will appear longer than tracheids. But is it actually? No. Why? Because in tracheids, one cell is representing one tube. But in vessels, many cells placed one above the other, common wall get dissolved, and then it makes a whole pipe-like, one tube-like structure. Clear? Chalo, read it. So, vessels are long cylindrical tube-like structures made up of many cells called as vessel member. And these vessel members are also sclerenchymatous, dead cells, devoid of protoplasm, cell wall lignified. Right? And they have a large central cavity called lumen, and they help in efficient water conduction. Presence of vessel is a characteristic feature of angiosperms. I told you. Now let me show you the arrangement of vessels in diagrammatic form. Are you all ready? So I told you that these vessels are dumbbell shaped. Each cell is small. They are placed one above the other. Suppose this is one vessel member. Dumbbell shaped cells. Another. Third. Fourth. So like this, these are vessel members that are placed one above the other. Now right now these are the plates. Till the time these plates, they do not vanish, how are they going to make one continuous pipe? So for that, these common plates, they dissolve, giving rise to whole pipe-like structure. So this is one tube that is made up of many cells, right? Coming to trachea, this is one tube that is made up of one cell. In case of vessel, this is one tube made up of so many cells. So you will feel that vessels are longer than tracheae, but are they actually longer? No, they are not longer, because in case of vessels, number of cells make one tube, and their wall is also thickened due to the deposition of lignin. They help in conduction of water and minerals. Clear to everybody? Nice. Very good. Moving ahead towards the third type of xylem tissues, that is xylem parenchyma. Now, xylem parenchyma, as the name suggests, these are those types of xylem components which are made up of parenchymatous cells. Now, since they are made up of parenchyma, my dear students, so will they be living or dead? They will be living in nature. Correct? And what will be their role? Surely they will not provide strength and support. What is the role of parenchyma? I told you, wherever parenchyma is present in the body, they will either help in storage or secretion. So xylem parenchyma, their prime function becomes storage or secretion. Yes or no? Clear to everybody? Now, some xylem parenchyma, they are also radially placed along the pith. Suppose this is a cross section of stem. Center, you know, always is made up of pith. Pith may be large, may be small, depends upon the condition. From pith arises in radial direction xylem parenchyma. Some cells of xylem that are parenchymatous, they arise in radial, sidewise direction, and these are known as medullary rays. Remember, in lateral meristematic tissue, I used this terminology, medullary ray. How many of you remember? If you remember this thing, then surely you are paying attention to the lecture properly. And if not, then please students, take the lecture, take the classes seriously. They are done for you. It is not giving me any benefit. All the things are done for the benefit of student. This victory batch, what is the purpose of launching this batch? Is it for our benefit? No. It is for your benefit. Now you are going to appear in NEET. And in NEET, because time left is very less, you need some lectures which are just in one shot, everything is covered which is important, right? So all these lectures which are given to you, they are important as they are one shot. Everything which is important for NEET is covered in these lectures. So please pay attention to these lectures, listen to them properly. You may take halves. No need to listen to the lecture in one go. You can take breaks, right? And then you listen to the lecture. But if you listen, then everything should go in your brain. Okay, coming back. So what are medullary rays? These are actually xylem parenchyma that are present in radial direction. And their function is conduction of water and mineral in radial direction. In radial direction. Clear? So these are certain roles of xylem parenchyma. Hope it is clear. This is the third component of xylem. Now moving towards the fourth and the last component of xylem, that is xylem fibers or xylem sclerenchyma. Again, these are the dead components. Why? Because the cell is devoid of protoplasm. Cell wall is lignified. Right? So lignified cell wall. Fourth, they are actually made up of which type of sclerenchyma? Name itself suggests they are made up of xylem fibers. Right? And if they are dead in nature, then their main role will be providing mechanical support or strength or rigidity to the plant body. Now, xylem fibers are long, elongated, tube-like structures with tapering ends. They may be septate or they may be aseptate. Aseptate means no cross walls are present, like in B. And they may be septate, that means some partitions are present in the cell. So this is very important point. Xylem fibers may be aseptate or septate. I hope xylem is now clear to everyone, which is one of the complex permanent tissue, very, very important. Moving ahead towards the next component of complex permanent tissue, phloem. Now, phloem are also conducting tissues. They are also found in pteridophytes, gymnosperms and angiosperms, but little bit different from xylem. How? First thing, phloem are responsible for carrying prepared food from source to sink. Right? Second, unlike that of xylem, these phloem are living components. Why? Because they are richly made up of parenchymatous cells. Right? Third difference, unlike that of xylem, these phloem can function bidirectionally. That is, usually they carry food from leaf to different parts. But when the condition becomes unfavorable, then food can be carried in reverse manner as well. The stored food which is present in either root or stem can be transported to the upward part of the plant. That is why I use the term source and sink. In case of phloem, you can never say that food is only transported from leaf to roots. No, it can be vice versa as well. That is why when you say the function of phloem, the better way to say is that phloem carries food from source to sink instead of mentioning leaves to roots. Correct? And hence we can say that phloem functions bidirectionally. I hope these points are clear. Karo, chalo. Let's see about phloem. So phloem transport food material usually from leaves to other parts of the plant. Usually, not always. Clear? Now, second point, they function bidirectionally. Right? Next point, they are living components of the complex of the plant body. And same as that of xylem, they are also represented by 4 subcomponents. Those are sieve tube cells, companion cells, phloem parenchyma and phloem fibers. Except phloem fibers, all are living components. Phloem fibers are the only dead component of phloem. Right? Now, if I talk about in gymnosperms and pteridophytes, they have phloem. However, in their phloem, instead of sieve tube and companion cells, they have sieve cell and albuminous cell. Presence of sieve tube and companion cell is a characteristic feature of angiosperms only. Very important point. Let's write. in gymnosperms, क्या चीज है gymnosperms में? Gymnosperms have sieve cells and albuminous cells instead of sieve tube and companion cells. The presence of sieve tube and companion cell is a characteristic feature of angiosperms. Right? Now, these are some points of phloem that you have to remember. As we did for xylem, we can also do for phloem. Like on the basis of origin, phloem again can be of two types. Primary phloem and secondary phloem. If I talk about primary phloem, again they can be of two types. What? Protophloem and metaphloem. Proto means initially formed phloem. They will have narrow sieve tubes, and then comes your later formed phloem called as metaphloem, which have wider sieve tubes. So I don't think you will forget it. The same manner we did for xylem, you have to do for phloem. Proto means initially formed. Meta means later formed. In case of protophloem, sieve tubes, the space within sieve tube is narrow. But in case of metaphloem, the sieve tubes are wider. Hence, again the same question. Which one will be better for conduction of food? Proto or meta? Then your answer will be meta for the same reason. Clear to everybody? The introductory part of phloem. Shall we move ahead? Yes, ma'am. Very good. Now, when we move ahead, we are going to talk about the components of phloem. Starting with the components of phloem, the first one is sieve tubes and companion cell. I am going to teach you both these components together. Why? Because functionally both these components are related. Originally also both these components are related. Sieve tubes and companion cells together, they are known as sister cells. Why? Why are they known as sister cells? Who are sister? Two girls can be called as sister if they have a common mother, right? So sieve tubes and companion cell, they have a common mother cell which divide to produce two different cells. Hence, sieve tubes and companion cell, they can be called as sister cell. Second point, if sieve tube is there, companion has to be there. If companion is there, sieve tube has to be there. So they are always found in association of each other. That is why we are going to study about both the components together. First, I am going to draw and then I am going to explain. Sieve tube. So number of sieve cells when placed one above the other, they form sieve tube. This is one sieve cell. Sieve cell, sieve means what? A perforated plate. In Hindi we call it as channi. So a cell whose above and below plate has holes. See like this, holes. So such plate is known as sieve plate. And a cell having sieve plate is known as sieve cell. When number of such cells are kept one above the other, then they form sieve tube. So this is sieve tube, which is absent in case of gymnosperms and pteridophytes. They only have sieve cells dispersed. But in angiosperms, these dispersed sieve cells, they come and form a proper tube. Right? Now when sieve cells of sieve tube, when they are young, they have a rich cytoplasm, a well-defined nucleus, everything. When these sieve cells are young, However, when the sieve cells they become mature, then on maturity they lose their nucleus, and along with the loss of nucleus they also lose all other cell organelles. So now that means we can erase this part, right? So sieve cells, initially they were living, but on maturity they tend to lose their nucleus along with other cell organelles like mitochondria, Golgi, etc. Now when they are, so indirectly you can say they have become dead. A cell without nucleus is actually dead. So indirectly you can call them dead. Now if they are dead, how will they help in conduction of food? So some cell has to come that controls the movement of food in these sieve cells. Hence they are always found in association with companion cells. Companion cells are rich parenchymatous cells that have a large centrally located nucleus which not only control the function of companion cell but also maintains the pressure gradient in the sieve tube cells. Now these two cells, they are connected with the help of pit fields or plasmodesmatal connections. So they are connected to each other. Their common wall is having some openings so that the two cells they can be joined. The cytoplasm of the two cells can be connected, right? So these are the companion cells which are having well developed nucleus. Companion cells are the sister cells to the sieve tube cells and they are always found in association. They are connected with the help of pits or plasmodesmatal connections. Why? So that this big sized nucleus can maintain the pressure gradient in the sieve tube cell for proper conduction of food. Clear? So what are sieve tube cells and companion cells? These are two important components of phloem which are actually responsible for the food movement. Sieve tube cells on maturity, they lose their nucleus. Hence, they are found in association with companion cell whose nucleus regulate the pressure gradient in the sieve cell. Companion cells are also parenchymatous cells and they are connected to sieve tube cell with the help of pit fields or plasmodesmatal connections. These two cells are sister cells because they have originated from the same mother cell. So these are certain points which you have to remember for sieve tube and companion cell. Hope you will remember, hope you understood this. Now, moving ahead with the third component of phloem, that is phloem parenchyma. Phloem parenchyma is also a living component, right? The name itself suggests this component of phloem is actually made up of parenchymatous cells. Now, if they are made up of parenchymatous cells, then what will be their function? Mainly storage or secretion. One more point you can remember, phloem parenchyma, they are absent in monocots. If you study the phloem of monocots, then they do not have phloem parenchyma. Moving towards the fourth component of phloem, phloem sclerenchyma, which are also known as bast fibers. These are the exceptions in phloem. They are the only component of phloem which are dead, as they are made up of fibers. Which fibers? Long elongated with tapering ends, lignified cell wall fibers. Now, if they are fibrous, they are dead, then they cannot help in conduction. They cannot help in storage or secretion. What will be their main role? Providing mechanical strength. They are not found in primary phloem, correct? So they are not found in primary phloem. They develop only when secondary phloem develops. That means monocots will again lack phloem fibers as well, right? Some economic importance phloem fibers are like jute, hemp. Jute you use for making bags and mats. So what is jute? Which plant product is jute? So jute are phloem fibers. They are bast fibers which have great economic importance. They are used for making mats, ropes, etc. So these are the points which you have to remember as well for phloem. So with this, the simple permanent as well as complex permanent tissue is over. Hope you have understood this chapter till here. Now we are going to move ahead and will start with the next concept in the chapter, that is tissue system. So now I hope all these components of the phloem is done and clear to everybody. Moving ahead with the next topic, that is tissue systems. But before moving towards tissue system, I can see on my screen there is one question. Let's try to solve this question. The question says, which of the following is incorrect for parenchyma? 4 statements are given to you and you have to select that which among the following statement is wrong for parenchyma. First, cells are isodiametric. Is it right? Yes, ma'am, it is correct. It makes majority of the plant body. Again, this is the right statement. Made up of dead cells. No, beta. Parenchyma are made up of group of living cells. So the incorrect statement in this case becomes C, and answer of the question also becomes C. Hope you are able to solve this question. If not, then revise the lecture again properly. Now, moving ahead with the next and important concept of tissue system. You have so far studied the types of tissue, meristematic as well as permanent. Now these tissues, they make up the whole plant body, and we have divided the whole plant body into 3 types of tissue system. The 3 types of tissue system are epidermal tissue system, ground tissue system, and vascular tissue system. Correct? Epidermal tissue system includes the outermost covering of the plant body, be it root, stem or leaf, and associated appendages. Vascular tissue system comprises of vascular bundles, that is arrangement of xylem and phloem. Now, apart from epidermis and apart from vascular tissue system, whatever is left in the plant body comes under ground tissue system. So tissue system overall, the plant body is divided into 3 tissue system, that is epidermal, ground and vascular, right? And this is with respect to all the organs of the plant body, be it flower, stem or root. So one by one we are going to see all the 3 types of tissue system. First, starting with epidermis. What do you mean by epidermis? Epidermis represents the outermost covering in the plant body of roots, stems as well as leaves. So epidermis and associated appendages forms epidermal tissue systems. It forms the outermost covering of the plant body, which is made up of single-layered, usually single-layered, compactly packed parenchymatous cells. Sometimes this epidermis may be multi-layered also. So first of all, epidermis is the outermost layer of the plant body, of primary plant body, which is usually single-layered, compactly packed cells made up of parenchyma, right? Usually single-layered, but sometimes, like in case of nerium, the epidermis in case of nerium is multi-layered. So this I'll write in bracket, that epidermis can also be multi-layered as seen in case of nerium. But usually it is single-layered. It is a protective covering. which is made up of compactly packed parenchymatous cells, elongated parenchymatous cells. So this is how epidermis is. It represents the outermost covering of the plant body, right? Now, usually in case of leaves, on the epidermis of the leaf, sometimes there is found deposition of a chemical called cutin, a waxy substance called cutin. This cutin avoids the loss of water vapor. It blocks transpiration. So this extra deposition of cutin is seen in the epidermis of leaf. However, the presence of cutin is absent in roots. You know the reason why? Because in roots there is no loss of water. There are no need of deposition of cutin, the waxy coating. Correct? So epidermis are parenchymatous cells. Yes. Now the outermost is usually covered with cuticle, that is cutin, to avoid the loss of water, but it is absent in case of roots. The main function of epidermis, the outermost covering, is providing protection. What is the main role of epidermis in epidermal tissue system? Obviously, providing protection to all the underlying tissues. It is the outermost covering. Even in your body, the first layer of your body is represented by epidermis. Then below you have dermis and all such layers. What is the function of epidermis? What do you study? Providing protection. It protects the layer of epidermis, protects all the tissues which are present below it. Right? Now, this epidermis also give rise to certain structures which are called as epidermal appendages. One by one, we are going to study all of them. In case of leaf epidermis, epidermis is usually continuous layer with no patches in between. However, in case of leaf epidermis, it is not so. In case of leaf epidermis, the epidermis is not continuous. Leaf epidermis is discontinuous. Can you tell me the reason? Can you guess it, students? Why it is discontinuous? The reason of leaf epidermis being discontinuous is the presence of stomatas. So stomatas are the minute openings found in the epidermis of the leaf that regulates the exchange of gases during processes like photosynthesis, respiration, and also control loss of water during transpiration. Now these stomatas, they are not found in root epidermis, not found on leaf epidermis. Hence their epidermis is continuous, compactly packed. See, it's a continuous layer, right? But exception is there, that is leaf epidermis. Now I am going to teach you what are these stomatas. One by one we are going to study all the extra structures found on epidermis. First, let's start with stomata. So what are stomatas? How will you define stomatas? These are the minute openings found on the epidermis of the leaf. What is their role? Their role is regulation of exchange of gases during photosynthesis and respiration. Plus, they also regulate the loss of water through these openings by the process of transpiration. Right? Now, suppose this is epidermis. Right? So, epidermis in case of leaf is discontinuous. You can see there are some openings present. So these openings, these pores are actually stomatas. Now these pores are guarded by specialized epidermal cells called guard cells. So in openings ke paas mein, these are the openings, right? Now the epidermal cells which are near to these openings, they get specialized to form guard cells. The name itself suggests these cells are responsible for regulating the opening and closing of stomata. Now I am going to show you the proper diagram of specialized guard cell along with the opening. See this. This is how the guard cell gets specialized. So these guard cells, they get specialized in this manner in case of dicots. In case of dicots, guard cells are the ones the way I have drawn, kidney shaped, right? Whereas in case of monocots, these guard cells are dumbbell shaped. The dumbbells which you pick in the gym. So they're exactly like dumbbells. And they are either in monocot or dicot, they are specialized epidermal cells. Here I am drawing kidney shaped guard cells which are observed in case of dicots. One thing, these guard cells, they regulate, or they're actually surrounding the opening which is called as stomatal pore. These are the guard cells, right? Now these guard cells in turn, they are surrounded by some more specialized epidermal cells that are known as accessory cells. So they are again protected. Guard cells are then protected by some more epidermal cells called as subsidiary cells or accessory cells. Correct? Now, pore plus guard cell plus subsidiary cell, together they are known as stomatal apparatus. So you can write one more thing, GC plus pore plus subsidiary cell, together they are known as stomatal apparatus. Clear? Will you remember this thing? This question was asked recently in the NEET examination. So just remember this thing. It may be asked. The question may get repeated this year as well. Okay. Now coming back to the diagram. Guard cells, the inner wall of the guard cell seems to be thick. This is the inner wall of guard cells. It seems to be thick. So the inner guard cell wall is thick in nature, whereas outer wall is thin. So this is the outer guard cell. So outer wall in guard cell is thin, whereas the inner guard cell inner wall is thick. You have to remember this thing. Guard cells, they also show radial arrangement of cellulosic fibrils which help in proper regulation of the opening and closing of stomata. So these are the radial microfibrils, radially arranged what? Cellulosic microfibrils, which helps in proper opening and closing of stomata. Epidermis, my dear students, being parenchymatous, they don't have chloroplast. However, the guard cells are the only ones which have chloroplast. So guard cells, apart from regulating opening and closing of stomata, they may also help in photosynthesis. Clear? Yes or no? So this is how the opening of stomata is regulated by guard cells. When guard cells are turgid, stoma, the opening opens. When guard cells are flaccid, the opening, the stoma, it closes. This concept of regulation will come in the chapter transport. But here you just have to focus on the structure. And what the structure says, there is an opening which is guarded by specialized epidermal cells having chloroplast and called guard cells, and then those guard cells are then covered and protected by some more epidermal cells called subsidiary or accessory cells. Subsidiary cells plus pore plus guard cell, together they are known as stomatal apparatus. Right? I hope this concept is clear. This much you have to do for stomata. Now moving ahead with more appendages of Epidermis, root hair. In case of root epidermis give rise to some unicellular elongations called root hair. Root hair are responsible for increasing the surface area for more absorption of water and mineral. You have studied in morphology from the zone of maturation, these root hair arises exogenously. So what are root hair? Root hair are the unicellular elongations or rather extensions. These are unicellular extensions from the epidermis of root that increases the surface area for better absorption of water and mineral. Increase the surface area for more absorption. You know, from the region of maturation, these root hair, they arises, and these are the extensions, these are the appendages found in the root epidermis. Root epidermis, my dear students, can also be called as epiblemma. So I'll write later, but now I'm telling you root epidermis, the other term for root epidermis is epiblemma. Coming to the next appendage of epidermis, which is found in case of stem, called as trichomes. These trichomes are the usually multicellular, sometimes unicellular, may be soft, may be stiff, may be branched, may be unbranched, may be secretory, may be non-secretory. Extensions, hair-like extensions found on the stem epidermis. So what are trichomes? Trichomes are hair-like outgrowths. Hair-like outgrowths found on stem epidermis. Hair-like outgrowths found on epidermis, which may be multicellular, unicellular, sometimes soft, sometimes stiff. If they are soft, then they have some different function. If they are stiff, then they can help in defense, may be branched, may be unbranched, they may be secretory, may secrete some chemicals, or they may not be secretory. So suppose this is my stem epidermis, compactly packed parenchymatous cells, right? From these cells may arise some hair-like outgrowths like this. So they may be like, this is branched trichome. Can you see I have drawn branched? This is non-branched. They may be soft or may be stiff, pointed. Then they will help in defense mechanism, right? They may secrete certain chemicals, they may not secrete. So this is how trichomes are. If they are stiff, then they provide protection for from the browsing animals. Correct? So these are some appendages of epidermis. Along with their appendages and the concept of epidermis, I hope the first tissue system, that is epidermal tissue system, is clear. Moving ahead towards the next tissue system, that is ground tissue system. Now, ground tissue system is actually the major tissue system that makes the major part of the plant body. Now, I'll just show you a simple diagram here to depict all the three tissue systems in a plant body. Like for example, if I cut a section of stem, dicot stem, right? So what I will find, now if I cut that section of dicot stem and try to view it under microscope, then what will I see? Layer by layer, let's talk. So the outermost layer will be of epidermis. Yes or no? And from this epidermis may come root hair, may come what? Hair-like outgrowths called trichomes. So this much is epidermal tissue system. Right? Now, below epidermis will be present what? One or two layered thick hypodermis. Below the epidermis is dermis or hypodermis. Below hypodermis, what you will see? Some few layered thick cells, compactly packed parenchymatous cells, which is called as general cortex. So this region is called as general cortex. The innermost layer of general cortex is known as endodermis. So next layer below GC will be endodermis. Suppose this is endodermis. Right? Below endodermis, then you will find layer of pericycle, few layered thick pericycle. Pericycle will enclose the centermost region of the dicot stem, which is called as pith. So whole this region, everything which I have written apart from ETS is GTS. This is differentiated ground tissue system having hypodermis, general cortex, endodermis, pericycle and pith. Right? Very good. Now this pith will be then surrounded by vascular bundles. That is your packets of xylem and phloem. Xylem and phloem, bundles of vascular bundles, they come under third tissue system, which is vascular tissue system. But now we are going to focus on these parts. One by one, I am going to teach you about what is hypodermis, endodermis, general cortex, pericycle and pith. After completing GTS, then we will move to vascular tissue system. Clear? Yes or no? Epidermal tissue system? Done. What are we going to study? Now we are going to study GTS. Clear? One point I am going to tell you, my dear students, please focus. The tissues which are present below endodermis, that means pericycle, pith and vascular bundles. Tissues present below endodermis comprises steel. So you can say the region present below endodermis is stellar region, steel region, and the region which is present above endodermis, this region, it is extrastellar region. Outside steel, inside steel. What is steel? Steel comprises of those tissues which are present below endodermis, and it includes pericycle, pith as well as vascular bundles. Clear what is the concept of steel? Yes, very good. Now let's start with ground tissue system specifically. So first of all, you should know ground tissue system. What is it? So ground tissue system, all tissues except epidermis and vascular bundle. I told you, remove ETS, remove VTS. Rest everything is GTS. Clear first point. Second point, ground tissue system is well differentiated into three subzones: hypodermis, general cortex and endodermis. Below endodermis includes pericycle and pith. This differentiation you found in dicot stem, you find in dicot root, you find it in and monocot root. However, monocot stem do not have differentiated GTS. They have uniform GTS. When I will teach you the anatomy of monocot separately, there you will see that monocot stems do not have differentiated ground tissue system. Otherwise, dicot root, monocot root and dicot stem, in all the three, ground tissue system is well differentiated. Clear to everybody? Yes. Now moving ahead with the first part of ground tissue system that is present below epidermis called hypodermis. So what is hypodermis? It is a layer found below epidermis. What is the role? Protection. Provide protection to the underlying tissues like general cortex and endodermis. Usually hypodermis is absent in monocots, but they are found richly in dicot stems. They are found in dicot stem. Dicot stem hypodermis is present, and their hypodermis is made up of collenchyma. Whereas in case of monocot stem, it is made up of sclerenchyma. So first point, layer found below epidermis, it is protective. Hypodermis is sclerenchymatous in case of monocot stem, whereas it is collenchymatous in case of dicot stem. In roots, it is not there. Roots lack hypodermis, stem have hypodermis. Monocot stem, sclerenchymatous. Dicot stem, collenchymatous. Clear? Function, protection. Where it is located? Below epidermis. So I hope hypodermis is clear. Moving towards general cortex. Now, what is there in general cortex? Coming to this. General cortex is usually few layered thick. Usually they are made up of parenchyma. So for general cortex, you just have to remember 2 points. Multi-layered, made up of parenchyma. Now if it is made up of parenchyma, what will be the role? Function will be providing storage region. So it may help in storage, it may help in secretion. So this is the main function of general cortex. Now, general cortex, the innermost layer of the general cortex is known as endodermis. Endodermis represent a single layered parenchymatous compactly packed cells. Correct? In case of roots, now I'm going to teach you endodermis separately for root as well as stem. I hope first point is clear for endodermis, single layered, compactly packed parenchymatous cells. In case of roots, endodermis is represented by parenchymatous cells, compactly packed, barrel-shaped, cylindrical cells are there, right? On these cylindrical cells, on the inner tangential and radial walls, right? On the inner tangential and radial walls, see, written, where it is. Okay, I'll write it. It's not written on the screen. I'll write it. On their inner tangential and radial walls, like this, radial walls and inner tangential wall, they have a deposition of some specific water impermeable substance called suberin. Right? So suberin is deposited on their radial and tangential wall. Now, what is the nature of this suberin? My dear students, it is water impermeable. Thus, through endodermis, water cannot pass. Here, everywhere, suberin is deposited. On these barrel-shaped cells, parenchymatous cells, suberin is deposited. Now, due to the deposition of suberin, water cannot pass symplastically. Like this, water cannot pass. Water has to pass through the spaces or outside the endodermis. So, due to the deposition of suberin, which is a water impermeable substance, endodermis do not allow symplastic movement of water. Do not allow symplastic movement of water. Clear? And This layer, due to the deposition of suberin, in case of root, endodermis, is known as Casparian layer. What it is known as? It is known as Casparian layer due to the deposition of suberin, which is a water impermeable substance. This is seen in case of roots. Now coming to shoot, that is stem. Stem, first of all, it is absent in monocot stems. Monocot stems do not have endodermis specifically. However, in case of dicot stem, endodermis stores, it is parenchymatous, and there the endodermal cells, they store starch grains. What they store? Starch grains. Hence, in case of dicot stem, the endodermis layer is also known as starchy sheath. So, due to the deposition of starch grains in the dicot stem, endodermis is known as starchy sheath. Clear? In roots, it is known as Casparian layer. In dicot stem, it is known as starchy sheath. And now you know the reason why. This much you have to do for endodermis. Moving ahead, below endodermis, all the tissues which are present are stele. So now let's start the first component of stele, which is pericycle. My dear students, pericycle are also few layered, thick, usually parenchymatous, sometimes sclerenchymatous cells present below endodermis. So what is pericycle? Once again, first point, it is a part of stele. Right? It is few layered thick, either parenchymatous or sclerenchymatous. In case of roots, they are parenchymatous. In case of stem, dicot stem. Why? Because monocot stem do not have pericycle. So in case of dicot stem, pericycle is sclerenchymatous. In case of dicot stem, pericycle is sclerenchymatous. So suppose in case of dicot stem, suppose this is a single layer of endodermis having deposition of starch grains, right? Now, below endodermis, what will be present? Pericycle. In case of dicot stem, this pericycle layer is sclerenchymatous and present in the form of semilunar patches. Semilunar means half moon shape. So this is how pericycle is found in case of dicot stem, and they are sclerenchymatous, present in the form of semilunar patches. Clear? Semilunar patches of sclerenchymatous pericycle is found below endodermis in case of dicot stem. When I talk about roots, in roots they are parenchymatous, that means they are living. Since they are living in case of roots, so they can give rise to lateral roots. In case of dicot and monocot roots, pericycle is the origination point of some root-like structures called lateral roots. So they give rise to lateral roots. Right? Everything will again come when I'll teach you anatomy separately. But here I am hinting you everything so that when we do the comparative study of anatomy, then there you can correlate with whatever I am telling you now. Coming to the last part of the GTS, that is pith. Pith is also known as medulla. Again, it is parenchymatous in nature. It represents the centermost core part of the plant body, also known as medulla, and it is, told you, parenchymatous. Now, mid-pith may be well developed in some cases, may not be that much well developed in some cases. You will come to know when we will study the comparative part of anatomy. I hope ground tissue system is clear in everybody's mind. All the sections of ground tissue system we have done. Now the third tissue system that is left is vascular tissue system. Vascular tissue system simply means arrangement of xylem and phloem. Vascular bundles means group of xylem phloem. How they are arranged, now that we are going to study in this system. Correct? So, on the basis of the arrangement of xylem and phloem, vascular tissues can be, in fact, vascular tissue system can be of two types, radial and conjoint. What is radial? Radial arrangement, first of all, it is seen in case of roots, both dicot and monocot. So radial arrangement of vascular bundle is seen in case of roots. What happens in this? When xylem and phloem present around the pith are located on different radii. See how? Suppose this is a section of dicot root, correct? You know all the arrangement of the tissues. Outermost layer will be epidermis. Roots, there is no hypodermis, right? From epidermis will come root hair. You know, right now only you studied about all the systems. No hypodermis, then followed by general cortex, endodermis, pericycle, and center will be pith. So this is pith. Around pith, what will be present? Vascular bundles will be present. Correct? Now, these vascular bundles will be present in radial manner. Radial manner means what? Xylem and phloem will be present on different radii. That means this orange one is xylem and this purple one is phloem. So, xylems are present on this radii, correct? But if you see about phloem, then phloem they are present on this radii. So, the radial arrangement means when vascular bundles are present separately or on different radii. Such arrangement is radial, which is seen in case of roots. Coming to stems and leaves, the other organs of plant body, that is stems and leaf. Arrangement is conjoint. Conjoint means what? When both the vascular bundles they are present on the same radii, like this. Xylem, phloem. So can you see they are present on the same radii? So that is called as conjoint arrangement. Same radii. Correct? Now, conjoint arrangement can be further of two types. Open conjoint or closed conjoint. Now, what do you mean by open and what do you mean by closed? Open arrangement means, see now, open conjoint type of vascular bundle means when there is a presence of cambium in each vascular bundle. First of all, what is conjoint? Same radii. So here is xylem, here is phloem on the same radii along the pith. Now, if in between one bundle, that is between one xylem and phloem, if there are group of actively dividing cells called intrafascicular cambium. Remember, lateral meristem, secondary meristem, interfascicular cambium. So, if intrafascicular cambium is present in each vascular bundle, if this cambium is present, then it is open type. Correct? Open type of vascular bundle. It is seen in case of dicot stem. Correct? Contrary to this is closed vascular bundle. Closed vascular bundles, closed conjoint are those in which there is no cambium. No cambium is found. That means simply xylem and phloem, they are present on the same radii, but they do not have such cambium present. And this type of arrangement you see in case of monocot stem, monocot leaf, as well as dicot leaves. Clear? In roots, radial vascular arrangement are always closed, be it dicot root, be it monocot root. In both the case, they are having radial arrangement, and in radial arrangement, there is no presence of intravascular cambium. So in case of root, irrespective of being dicot or monocot, their system is always closed. So with this, the third tissue system and the concept of tissue system is done. I hope you have understood ETS, GTS and VTS well. With this, the concept of tissue system, the different types of tissues are done, and now we are moving ahead with the next topic of the chapter, that is anatomy of different dicot, monocot root, dicot, monocot stem, dicot, monocot leaf, comparative study of all these three structures. Okay? Are you all ready, guys? Very good. Now, here you can see I have drawn it in the form of a chart. So yes, we are going to do the comparative study in the form of chart only. First, starting with dicot root and monocot root. Major portion I have already told you while I was teaching you the tissue system. You know what is epidermal tissue system, you know what is ground tissue and what is VTS. You just have to separate them and sum them according to dicot and monocot root. If you cut the section of the root, be it dicot or monocot, what anatomy you will see? Outermost layer epidermis, hypodermis is absent, cortex, endodermis, pericycle, bundles, and then pith. Same anatomy you will see. So same thing you can write it over here. So outer to inner, what will you see? Both in dicot and monocot root. Epidermis, one thing. No hypodermis. Then next will be general cortex. Then endodermis. Then pericycle, then vascular bundles, and last will be pith. So these are the regions which are found in the anatomy of root, both dicot and monocot. Now one by one, write about all the things. Epidermis, common. What will you write? Outermost covering, single layered, parenchymatous, and give rise to unicellular root hair, which is common for both dicot as well as monocot, no difference. So one I am going to write, second you can copy it. So it is unilayered, parenchymatous, compactly packed, and give rise to root hair. The other term for epidermis is epiblemma. No cutin. I already told you, you know this. No cutin. In roots, no cutin is required. No loss of water is taking place. Coming to general cortex. Common for both monocot and dicot. Same. Few layered thick, parenchymatous. Few layered thick, made up of parenchyma, hence helps in storage and secretion. Correct? Same. No difference. Same, both in dicot and monocot root. Coming to third, endodermis. Same. What? Made up of barrel-shaped cells, single-layered, parenchymatous, Casparian, deposition of suberin on the radial and tangential wall. So simply I am going to write one term, Casparian layer. Now you know this. No need to write further. You know it. Just 5 minutes back only I explained you everything. Coming to the next, pericycle. Again, pericycle is few-layered parenchymatous in case of roots. So pericycle, few-layered parenchymatous, give rise to lateral roots. Yes or no? We wrote this concept. Give rise to lateral roots. Correct? Very good. You know this thing, already done. Next, vascular bundles. So vascular bundles in case of root. Again you did it. Radial vascular bundle means closed. Always radial. If they are radial, then they are automatically closed. The number of vascular bundle in case of dicot root, they are limited, that is 2 to 4. Here the difference come. In case of monocot roots, my dear student, the number of the arrangement is same, radial only. But the number of vascular bundles, they are more than 6. And this condition is known as polyarc. Polyarc condition, when there are more than 6 vascular bundles. So in case of monocot roots, the number of vascular bundle are more as compared to dicot roots. Clear? Coming to pith. Pith is there, but in case of dicot root, pith is small and inconspicuous, not that well developed. Parenchymatous, but not that well developed. But when we come to monocot root, here the pith is large and well developed. So comes the second difference. Inconspicuous, small, here it is large and well developed. Clear? Two differences you saw, one on the basis of the number of vascular bundles and second on the size of pith. Third difference is monocot root, my dear students, do not show secondary growth. No formation of lateral meristem. But in case of dicot root, they show secondary growth. They undergo secondary growth. How? Their cambium is absent. No cambium. They have closed vascular bundle. No cambium. Then how secondary growth? So during the time of secondary growth, pericycle in case of dicot root, not in monocot root, performs some extra function. The cells of pericycle undergoes dedifferentiation to form the cambium. Right? So this is extra point in dicot root that you have to remember, that in case of dicot roots, pericycle, apart from giving rise to lateral root, also help in the formation of vascular cambium. So this is extra. So I'm writing it with some different color. Let me write it. So they give rise to vascular cambium as well. And it is an exclusive feature of dicot root only, not seen in monocot root. Therefore, monocot root shows no secondary growth, but dicot root shows secondary growth. Clear? So this much you have to do in the anatomy of dicot and monocot root. Just once read it from NCERT. That's it. And your concept will be done. I hope it is clear. It is very, very simple. If you have listened to me properly when I was teaching you tissue system, then this comparison is going to become very easy for you. Yes? Clear? Now, moving ahead. Now we are going to see the anatomy of dicot stem and monocot stem. Starting with dicot stem. Again, when you cut the section of either dicot or monocot stem, what are you going to find? Now here there will be a difference. You know, in case of monocot stem, ground tissue system is undifferentiated. But here in case of dicot stem, ground tissue system is well differentiated into 3 subzones. Right? Yes or no? Chalo. So let's see now. Okay. So first, outermost both in dicot as well as monocot stem will be epidermis. In case of stem, hypodermis is present both in dicot and monocot. Now, below hypodermis will be general cortex both in dicot and monocot stem. But now, in case of monocot stem, everything will be general cortex. Now, general cortex is not further differentiated. Here, in case of dicot stem, general cortex, endodermis, pericycle, pith, everything is found. Clear? So first, we are going to study those points which are common both in dicot as well as monocot stem, that is epidermis and hypodermis. So both in dicot and monocot stem, epidermis is single layered, parenchymatous, compactly packed, may give rise to hair-like outgrowth called trichomes. You already know the concept of stem epidermis, right? So here you can write in epidermis, it is single layered, parenchymatous, give rise to hair-like outgrowths called trichomes. Right? Now, coming to hypodermis. Hypodermis is found in both monocot and dicot stem. But in case of stem, dicot stem, hypodermis is collenchymatous. May be present continuously or in the form of patches. However, in case of monocot stem, hypodermis is sclerenchymatous. So this is one major difference that you see. Correct? Now, coming to dicot stem, in case of dicot stem, general cortex is few layered thick, parenchymatous. Now, monocot stem I will do separately. Few layered thick and parenchymatous. Correct? Endodermis, you know, it is known as starchy sheath. Already told you, starchy sheath, which is absent in case of monocot stem, starchy sheath. Pericycle, again you know, sclerenchymatous, present in the form of semi-lunar patch. I have shown you with the help of diagram as well. So pericycle, here you can write sclerenchymatous, present below endodermis in the form of semi-lunar patches, half moon shaped. Correct? Absent in case of monocots. Absent. Right? I'll show you for monocot stem what you have to write for general cortex. Coming to pith. Pith, here you can write it is present in the center, made up of parenchyma. Correct? Present in the center, parenchyma. From the pith arises medullary rays which further undergoes dedifferentiation and help in secondary growth. Here you will write absent. Now talking about vascular bundles. My dear students, in case of dicot stem, vascular bundles are conjoint and open type. So vascular bundles here, they are conjoint as well as open type. Now, if I talk about monocot stems, then vascular bundles, they are conjoint but no cambium present. So they are closed type. Plus, in case of monocot stem, the vascular bundle, they show dimorphism. Now, what is this dimorphism? I'll show you with the help of the diagram. Dimorphism means two types. Di means two. So, in case of monocot stem, the vascular bundle shows dimorphism. Now, what is this dimorphism? Let's see. So, I'll do it with the help of a diagram. Here, I'm going to draw a section of monocot stem. a circular section of monocot stem. So now just see this. Right? So outermost layer will be epidermis. Inner to epidermis will be hypodermis, which will be sclerenchymatous in nature. So this is what? Hypodermis, that is sclerenchymatous. Let me write it in brackets so that you can remember it again. Now, all the tissues now which are present inside, everything is coming as general cortex. Because in case of monocot stem, it is not well differentiated. The ground tissue system is not well differentiated. So everything which is enclosed within hypodermis comes under as general cortex. No endodermis, no pericycle, no pith. Now, in this general cortex, vascular bundles are scattered. One more point I remembered. In case of dicot stem, my dear students, arrangement of vascular bundle is ring-like. Vascular bundles are arranged in ring-like manner. However, in case of monocot stem, vascular bundles are scattered in the general cortex. Correct? Okay. Now, see this diagram. So, general cortex, here vascular bundles are scattered. Vascular bundle shows dimorphism. That means the vascular bundles which are present towards the periphery, they are compactly packed and small in size. Like this. So, the vascular bundles in case of monocot stem which are present towards the periphery, towards the hypodermis are, first of all, closely packed, plus they are smaller in size. Correct? But if I talk about the vascular bundles which are present in the center, they are large and loosely packed. Right? So, vascular bundles which are present in the center, they are large and loosely packed. They are conjoined and closed type. Here also conjoined closed, here also conjoined closed. That means xylem and phloem, they are lying on the same radii and there is no cambium present. Correct? But they are showing dimorphism now. This is one type and this is another type. So in case of monocot stem, vascular bundles, they show dimorphism. They are scattered in the general cortex. Dimorphism means two types. Some are compactly packed, some are loosely packed. The ones which are compactly packed are present towards the periphery, and the ones which are present in the center, they are first of all loosely packed and they are larger in size. So I hope this point you are going to also remember for monocot stem. So their vascular bundles are conjoined, closed, showing dimorphism, scattered. But in case of dicot stem, vascular bundles are conjoined, open because they will undergo secondary growth and arranged in ring-like manner around the pith or medulla. So this is what you have to study for the anatomy of dicot and monocot stem. Are you able to analyze the difference? First, epidermis to same hai. Hypodermis composition is different. Cortex, if I talk about cortex, then in case of dicot stem it is differentiated. In case of monocot it is undifferentiated. Every region, everything which is present under hypodermis is general cortex. But in case of dicot stem, general cortex is few layered thick parenchymatous, below which is endodermis, that is starchy sheath. Why starchy sheath? Due to the deposition of starch grains. Below is pericycle, which is sclerenchymatous and present in the form of semilunar patches. Sclerenchymatous and present in the form of semilunar patches. Then comes the vascular bundles and then come the pith. I hope this whole anatomy of dicot root, monocot root, like this over here, dicot root, monocot root, and dicot stem and monocot stem is clear to each and every student. With this, we are over with root and stem. Now we are going to head towards the anatomy of dicot and monocot leaf, called as dorsiventral and isobilateral leaf. Now we are going to start with the last segment where we are going to compare the anatomy of dicot leaf and monocot leaf. I hope the remaining part of the chapter which we have covered so far is clear to everybody. Now moving ahead with the next segment of the chapter where you are going to compare the anatomy of dicot leaf and monocot leaf. So let's start without wasting the time. Dicot leaf, which is also known as dorsiventral leaf. Dorsiventral leaf. Whereas monocot leaf is also known as isobilateral leaf. Now, what do you mean by dorsiventral and why dicot leaf is known as dorsiventral? So, first point is that in case of dicot leaves, usually their arrangement is how? It is like this, right? So, that means they are almost placed perpendicular to sunlight. Now, since the dicot leaves, they are placed perpendicular to sunlight. So, the upper part of the leaf receives more light as compared to the lower part. Now, since the upper part of the leaf is receiving more light, that means it will develop more amount of chlorophyll so that extra amount of light can be easily trapped, and lower part of the leaf will be comparatively having less amount of chlorophyll, because of which you must have observed in dicot leaf, upper side of the leaf is greener, is more greener, is dark greener as compared to the lower side. You turn the leaf, you find some light coloration of green, but the upper side is even darker than that. So that means in case of dicot leaf, there are two distinct sides, the dorsal side and the ventral side. You can clearly distinct the two sides of the leaf on the basis of color. So that is why dicot leaves, as they can be easily distinguished, the two sides of the leaf can easily be distinguished on the basis of color. That is why these leaves are known as dorsiventral leaf. So why they are known as dorsiventral leaf? Due to the distinction in the sides of the coloration. So dicot leaves have clear-cut distinction in the coloration of leaf. First point that you have to know, in the coloration of leaf, that is, upper part of the leaf is greener as compared to the lower. My dear students, upper part is known as adaxial. We will use the term adaxial, whereas for lower we use the term abaxial. B for below, B for abaxial. So just remember it like this. But when you come to monocot leaves, then my dear students, monocot leaf are perpendicular to the sun. As a result, both the sides, they receive equal amount of light. Hence, in case of monocot leaf, like for example banana leaf, if you observe, then the color of banana leaf on both the sides is same. There is no distinction of dorsal side and ventral side on the basis of color. They both are almost of same color. Hence, monocot leaves are also known as isobilateral leaf. So, in case of monocot leaf, both the sides of the leaf have same coloration. Both the sides of the leaf, that is dorsal as well as ventral side, has same coloration as both the sides of the leaf receives the same amount of light. Now, this is about the coloration. Now, if we talk about the anatomy of both dicot and monocot leaf, then overall the leaf anatomy is divided into three regions, that is epidermis, mesophyll tissues and vascular bundles. So epidermal tissue system is there containing stomatas, then vascular tissue system is there, and in term of ground tissue system, leaves they only have mesophyll. So in case of leaves, ground tissue system is represented by mesophyll cells. So this point you have to remember, first thing. Now, moving ahead, as I told you, in case of leaf, the whole body is divided into three parts. Epidermis. Now, epidermis can be upper epidermis as well as lower epidermis. In between the upper and lower epidermis are present the mesophyll cells, and in the mesophyll cells, here and there are present vascular bundles. Right? First, we are going to talk about the epidermis, then mesophyll, and then vascular bundle. One by one, we are going to talk about all the three regions in the leaf. So first, if I talk about epidermis, then both dicot and monocot have one upper epidermis and lower epidermis. Now, what is the difference then? In case of dicot leaf, upper epidermis either have very less or no stomata. They are covered by cuticle. Lower epidermis have numerous number of stomata and they are also covered by cuticle. So in case of dicot leaf, upper epidermis, adaxial surface has less or no stomata. Lower epidermis have numerous number of stomatas, and both the surfaces they are covered with the help of cuticle. Correct? So this is the second point which you have to write below. Dicot leaf. Epidermis. Upper epidermis has more number of stomata as compared to lower epidermis. The number of stomatas are more in case of lower epidermis. It's lower epidermis as compared to upper epidermis. Talking about in monocot leaf, my dear students, number of stomatas are equal both in the lower as well as on the upper epidermis. Again, that is why their name is isobilateral. Color is also same and distribution of stomata is also same. Both on upper and lower epidermis, number of stomatas are same. So in case of monocot leaf, both the epidermis can contribute in almost equal amount of transpiration. But when it comes to dicot leaf, lower epidermis will be more active in transpiration as compared to upper epidermis. Clear? Very good. Now coming below the epidermis is the mesophyll tissues, that means parenchymatous cells. Now my dear students, in case of dicot leaf, mesophyll tissues are differentiated into two types. Some are elongated and some are spherical shaped. Towards the epidermis, you will find some elongated, compactly packed, tube-like parenchymatous cells called palisade parenchyma. Whereas towards the lower epidermis, the mesophyll tissues are represented by spherical or oval shaped, not so compactly packed cells called spongy parenchyma. So, in case of dicot leaf, mesophyll is differentiated into upper palisade parenchyma, whereas below spongy parenchyma. Right? This distinction is absent in case of monocot leaf. They only have one type of mesophyll cells which are spongy parenchyma. So they only have spongy parenchyma. They lack distribution, they lack differentiation of mesophyll tissues. Can you see the difference? Yes. Next, now in the leaf, you know there is a midrib and there is venation arrangement of veins and veinlet. So usually in case of dicot leaf, you will see reticulate venation. This point you already know. They have reticulate venation. But when we come to monocot leaf, they show parallel venation. Yes or no? So they show parallel venation, whereas in case of dicot leaf, there is reticulate venation. I hope the differences you are able to understand. Now moving ahead, with the help of diagram, let's try to analyze. So this is a dicot leaf and this is the monocot leaf. Now how did I understood? I did not mention that which is dicot and which is monocot. How I came to know? Simple, by seeing the differences. In case of dicot leaf, you can easily see there are two types of what? Mesophyll cells. Yes or no? See, the upper are long, elongated, compactly packed, green colored parenchyma, which is known as palisade. And towards the lower side, they have what? They have some spherical shaped, not so compactly packed, green colored cells called spongy parenchyma. And if I talk about in monocot leaf, they only have one type of mesophyll cells, that is spongy parenchyma. So this is one of the reasons that how I came to know that which is dicot and which is monocot. Clear? Right? Now, moving ahead, let's see the arrangement of their vascular bundles. So both in monocot and dicot leaf, vascular bundles are conjoined. That means xylem and phloem will be present on the same radii and they will be closed. Closed means no cambium, no secondary growth, irrespective of dicot or monocot. Dicot leaves also do not undergo secondary growth. So in both the cases, vascular bundles are conjoined and closed. Yes or no? So this is common in both the cases. Now there's some special point for monocot leaves. In monocot, like for example in grasses, the adaxial, the epidermis, that is the adaxial epidermis, some of the adaxial cells, adaxial epidermal cells in grasses, they modify to form some empty, colorless, large cells called bulliform or motor cells. These bulliform or motor cells, they are responsible for maintaining the turgidity or flaccidity in the cell. If the bulliform cells, they are turgid, then the leaf is wide open. But when these bulliform cells, they tend to lose the water, they become flaccid, then they curl the leaf inward. It is one of the modification of grasses to avoid unnecessary loss of transpiration during water stress conditions. So this point you have to remember, it is specially for monocot leaves like in grasses. So I am writing it, please pay attention. Questions are frequently asked on bulliform. So the entire concept of bulliform you should know. So in grasses, some adaxial or abaxial, adaxial, some adaxial epidermal cells, right, modifies to form bulliform or motor cells. Bulliform cells. These bulliform cells, my dear student, they are colorless, empty and large cells. Now, there are two conditions. When bulliform cells are turgid, in that case, the leaf is open. However, if they are flaccid, then in that case, leaf curls inward to avoid any loss of water due to transpiration. I hope this concept of bulliform cell is also clear to everybody. Yes. Now, this much is what you have to do in the anatomy of dicot leaf and monocot leaf, and this is how you are going to identify from the diagram that which is dicot and which is monocot leaf. With this, the comparison in the anatomy is also over. Now we are going to start the last but the very important section of the chapter, which is secondary growth. Now, my dear students, this is actually the section where you have to understand. The part of the chapter which we have done so far, they are basically understanding as well as learning is more. But here in secondary growth, if you understand the concept, then no need to learn. Just revise it once and everything will strike in your mind again. Right? So please pay attention and please listen to this concept very calmly and very easily. Just relax, lie down on your bed, keep your earphones on and just listen to it properly. Correct? Okay. So let's start with secondary growth. You have already learnt about primary growth. What is primary growth? Increase in the height of the plant body due to the presence of apical meristem and intercalary meristem. Intercalary and apical meristem are the two types of primary meristematic tissues which divide, produce new cells and gets differentiated to form primary permanent tissue. So primary meristem and primary permanent, together they are responsible for the primary growth of the plant body. In primary growth, height of the plant increases. However, the thickness of the root and shoot is not that much. Now, when it comes to monocots are like usually annuals and biennial plants, right? So even if their stem and if their roots are not well developed, they are not quite thick, then too they can manage, they can survive. As in monocots, they are not usually tall trees, they are small herbs and shrubs, they are limited to herbs and shrubs. So in them, primary growth is okay. Monocots do not need secondary growth. They need not to increase the thickness of their stem and root. But what to dicots? In dicots, majority of the plants are big trees. Correct? Now, if dicots you have seen huge big trees, and in such huge big trees, if the stem and root is thin, will those trees be able to survive? The wind will come and in 1 second they will fall off. But imagine if the stem and root is thick, then whatsoever wind velocity may be, the trees will stand erect in their position. So in that case, secondary growth becomes must. So in dicots of angiosperm, apart from primary growth, secondary growth also takes place in order to increase the thickness of their root and shoot. Clear? What is the purpose of secondary growth? ठीक. How? Primary permanent tissues tak to you know. Now when in dicots, when there is a need of secondary growth, these primary permanent tissues like parenchyma, they undergo de-differentiation. They de-differentiate, they regain their capacity of division to form lateral meristem, which is a type of secondary meristematic tissue. These lateral meristem, they arise on the sides of the shoot and the root, not on leaves, flowers or fruits, only on shoot and root. Once lateral meristem is formed, then they are meristematic, they cut, they divide, they produce new cells, and those new cells again they get redifferentiated to form secondary permanent tissues like secondary xylem, secondary phloem, secondary medullary rays and etc. So formation of secondary meristem and formation of secondary permanent is secondary growth, and it is restricted only in those plants who need thick stem and thick root, like in dicots. Clear? Secondary growth का होना is not 100% assurance of all plant. No. It is only seen in those plants who require it. And due to the presence of secondary growth, the plant growth becomes indeterminate, indefinite. You have studied this point when I was teaching you living world. You remember, growth in case of plant is indefinite. Why? Because of the presence of secondary meristematic tissue, lateral meristem. Every year, the thickness of the root and shoot keeps on increasing. These secondary meristem are active. Once they are formed, now they will remain active throughout the lifespan of the tree. That is why height is constant in a plant. That is a part of primary growth, which is constant, which is fixed. But thickness of the root and shoot is not constant due to secondary growth, due to lateral meristem. And that is why plant growth is known as indefinite or indeterminate growth. Clear? So highlighting points are clear. Now these points are written on the screen. I will read it for you. These points I have directly taken from NCERT so that along with the concept your NCERT is also revised. My majority of the points which I write on the screen are directly from NCERT. The only reason is that along with the concept, you also get to see the lines of NCERT. If you read the notes, you will see they are directly from NCERT. My only aim, my only purpose is that if you don't find time for reading NCERT, then I will let you do it. I will not let any of my student to go in the examination hall without reading NCERT. If you read my notes, you will automatically cover your NCERT as well. Correct? Now, coming back to the topic, secondary growth. Secondary growth, the growth of the root and stem in length with the help of apical meristem is called as primary growth. Correct? Primary growth is done, which is fixed. However, apart from primary growth, in dicot plants exhibit increase in thickness, in girth of root and shoot. This increase is called secondary growth. The tissues that involves in secondary growth are lateral meristem. And lateral meristem के examples दो हैं. There are two examples of lateral meristem, that is Vascular Cambium and Cork Cambium. Vascular Cambium intrafascicular and interfascicular. You already know these points when I taught you lateral meristematic tissue. I hope the introductory part of secondary growth is clear in each and everybody's mind. Very good. Now let's start with the main process of secondary growth. First, we are going to study secondary growth in dicot stem, and then I'm going to teach you secondary growth in dicot root. Correct? So now let's see secondary growth in dicot stem. Before starting the secondary growth in dicot stem, you should know the arrangement of tissues in dicot stem. So if I cut a circular section of a dicot stem, so you already know the anatomy of dicot stem as well. You have already studied it. The outermost layer is epidermis, which is associated with trichomes, together form the epidermal tissue system. Correct? Followed by epidermal tissue system is ground tissue system that comprises of hypodermis, then general cortex, then starchy sheath endodermis, then semilunar patches of sclerenchymatous pericycle, and then center is pith. Right? Yes or no? Am I saying it correct? Very good. And around this pith are present vascular bundles which are conjoined, open type and arranged in a ring-like manner. So around the pith, vascular bundles are arranged in ring-like manner. And the arrangement is conjoint. Conjoint means xylem and phloem, they are present on the same radii like this. So this is conjoint. And open type means within each bundle there is already presence of some actively dividing cells called intravascular cambium. Intravascular cambium is a part of lateral meristem, but it is primary in origin. It is an exception. You already know this. Question also you have done on this concept. Intravascular cambium is lateral meristem, helps in secondary growth, but still it is primary in origin. So this is your intravascular cambium. Correct? Now, in between each vascular bundles there are from the pith, from the medulla. Pith is also known as medulla. Right? From medulla, some parenchymatous cells arises in radial direction. Those are known as ray parenchyma or medullary rays. These medullary rays are responsible actually for the radial conduction of water and mineral. These are parenchymatous. So this is originally is how the arrangement of anatomy in dicot stem when the growth is still primary. Now a time comes secondary growth is required. For that, lateral meristem has to be produced by the process of dedifferentiation of primary permanent tissues. So my dear student, first step in dicot stem secondary growth is, in case of dicot stem secondary growth, first step is formation of cambial ring. So first you are going to complete the cambium. As you can see in the diagram, still the cambium is incomplete now. See, partial ring is there. Now we need to complete this ring, and for completing the ring, what happens? The cells, the parenchymatous cells of medullary rays which have lost their capacity of division regain their capacity. They undergo dedifferentiation to form interfascicular cambium. Medullary rays, the cells of medullary rays are then replaced by some actively dividing cells called interfascicular cambium. First point. Now these interfascicular cambium, they are lateral meristem. And which type of lateral meristem? Vascular cambium. Now here what will come now? Instead of medullary rays, interfascicular cambium will come in between each vascular bundle, thus completing the vascular cambial ring. So 50% of the ring is already present in the form of intrafascicular, but 50% of the ring arises later on by the dedifferentiation of medullary rays called interfascicular cambium. Now, this interfascicular cambium along with intra completes the cambial ring. Correct? So, what is the first step when you start the secondary growth in dicot stem? The first step is formation of cambial ring. 50% is already there. So, first step, see, in young stem, the vascular cambium are present in, the cambium is present in between xylem and phloem, each bundle, but there are 50%. So in order to complete the ring, what is required? The parenchymatous cells of medulla, medullary ray cells undergo dedifferentiation. Undergoes dedifferentiation to complete the ring. And do you know how? Yes, ma'am. 50% of the ring was already present. 50% of the ring, that is intrafascicular cambium, is already present, but remaining 50% of the ring, that is intervascular cambium, arises after the activity of medullary rays. Now, this inter and intra together completes the vascular ring, which is circular in outline. So, next point is that ring is circular in outline. Correct? See this in the diagram. Is it not circular in outline? Yes. Now, the first step is done. Second step is activity of this cambium. If this lateral meristem, if this vascular cambium is produced, then what is its role? Its role is to divide and produce new cell and let them redifferentiate. So now, this is cambial ring. Inner to cambium is primary xylem and outer to cambium is primary phloem. Now, this cambial ring is going to undergo division. It is going to produce new cells. Some cells are going to deposit on the inner side of the vascular ring and some cells are going to get deposited on the outer side of the vascular ring. The cells which deposit on the inner side, they redifferentiate to form secondary xylem, whereas the cells which redifferentiate or which deposits on the outer side of the vascular ring, they redifferentiate to form secondary phloem. So like this, you can say that secondary xylem is formed inside the cambium and secondary phloem is formed outside the cambium. Now my dear students, initially when secondary growth did not start, only cambial ring was there. So can you see, cambial ring is directly in contact with primary xylem and primary phloem. Tick. Now if I am saying that after this cambial ring is going to function, it is going to produce new cells, it is going to deposit secondary xylem and secondary phloem. Where is the space to deposit secondary xylem and secondary phloem? Where the primary xylem and primary phloem are in close proximity with the cambium. Suppose this is cambium. Tick. On the inner side is primary xylem, consider this finger, and on the outer side is primary phloem. So where is the space for the deposition of new cells? Now what will happen when this cambium is going to function and produce new cells on the inner as well as on the outer side? The already existing primary xylem and already existing primary phloem, now they are going to move apart. Now the distance between them is going to increase. Primary xylem will move towards the pith and crush the pith, whereas primary phloem, this will start moving on the outer side, damaging the cells which are outer to primary phloem, that is pericycle, endodermis, epidermis. Now they are going to damage the cells. Yes or no? Primary xylem will damage the pith. It is going to crush in the pith. But primary phloem is going to crush. It is going to move out and it is going to displace and damage the cells which are present outer to primary phloem. And what is present outer to primary phloem? The entire anatomy. All epidermis, hypodermis, cortex and endodermis, pericycle. All those. So primary phloem, due to the activity of vascular cambium, due to the activity of vascular cambium, primary phloem is going to damage the extrastellar region. Right now we are talking in stele region, na? We are talking in cambium only, within vascular bundles and pith. But due to the stellar activity, due to the activity of vascular cambium, the extrastellar region, that is the region outside the pericycle, endodermis, cortex, they all get damaged. Clear or not? Okay, once again I am telling you, pay attention. So what is the first step? First step is formation of cambial ring. I hope this first step is clear. Second step is what? Activity of the cambial ring. So what have to be done? This is dicot stem anatomy, right? Pay attention, very important. Outermost is epidermis. The centermost is pith, right? Around the pith are present number of vascular bundles arranged in ring-like manner, conjoint and open. Open due to the presence of intravascular cambium. Now, due to the activity of medullary rays, intervascular cambium is also produced, thus completing the vascular ring, thus completing the vascular cambial ring. Correct? Now, once this vascular cambial ring is formed, what it is going to do? It is going to deposit new cells on the inner side that are going to redifferentiate to form secondary xylem, and new cells on the outer side that are going to redifferentiate to form secondary phloem. As a result, the primary phloem will crush towards the pith, whereas these primary phloem, they are going to move towards the outer side, thus damaging the outer anatomy. First thing, correct? Now, the activity of this vascular cambium, the activity of vascular cambium is more on the inner side than the outer side. So, there is more deposition of secondary xylem as compared to secondary phloem. Correct? Shall I write all these points or you will do it? So, I'm just writing the highlights and then later you can read it. So, number 1 is cambial ring is complete. Is circular in outline. First important point. Question will be asked on this basis. Second, cambial ring produces new cells, some of which deposit on the inner side. The ones which deposit on the inner side, they redifferentiate to form secondary xylem, and some are deposit on the outer side of the cambium, which redifferentiate to form secondary phloem. Activity on the inner side is more. There are more deposition of cells on the inner side as compared to outer side. Now, due to the deposition of secondary phloem, primary xylem will crush towards the pith. Correct? It need to give space. So it is going to crush towards the pith. But the secondary phloem is going to damage, due to the deposition of secondary phloem, primary phloem is going to damage the remaining anatomy. Okay? Clear? Now what will happen? Stellar region activity is done. Now, due to the activity of vascular cambium, the cells which are present in the extrastellar region, that is cortical cells and epidermal cells, as well as your hypodermal cells, they have got damaged. So, some healing has to be done over there. Hence, there is now a formation of second type of lateral meristem in the extrastellar region due to the dedifferentiation of cortical cells called Cork Cambium. So now what will happen? Now primary phloem will damage the cells. This is epidermis. Below epidermis there is hypodermis. And then below that is again cortex. Right? Correct? So this is epidermis, hypodermis, and then this is your cortex. Now what will happen when primary phloem will move out? It is going to damage the cortical cells and then even epidermal cells. So in order to heal them, what will happen? These cortical cells, just let me remove this hypodermis. Oh, chalo, let it be then. These cortical cells which are parenchymatous, they undergo dedifferentiation to produce what? To produce another lateral meristem called Cork Cambium. So now here Cork Cambium will arise. The other term for Cork Cambium is phylogen. So how cork cambium is produced in the extrastellar region? Cork cambium, also known as phylogen. This phylogen develops by the dedifferentiation of cortical cells. And why it has come up? It has come up so that the damage created by primary phloem can be healed up. Now, phylogen is produced. This is actively dividing. Now, this cork cambium cells are also going to divide to produce some cells on the outer side and some cells on the inner side. So now this yellow colored cork cambium is going to cut new cells, produce new cells. Those new cells, some of the cells will deposit on the inner side to heal the injury of cortex. These cells which deposit on the inner side, these cells are known as secondary cortical cells. These secondary cortical cells are parenchymatous in nature. Right? Whereas on the outer side, they deposit some sclerenchymatous, suberonized sclerenchymatous cells called cork cells. The other term for cork is phellum. These are sclerenchymatous in nature. Correct? So now finally, due to the deposition of secondary cortex, which is also known as phyloderm, as well as cork cells. Cork cells are the cells which deposit on the outer side for healing the damage in epidermis. These cork cells are sclerenchymatous in nature. Now, in order to heal the injury of inner side, that is cortical cells, then there is a deposition of secondary cortex, also called as phyloderm, and these are parenchymatous cells. And like this, whole injury is healed up. New deposition of tissues has also been done, and that led to the increase in the thickness of the stem. Clear? This is how every year secondary growth takes place in case of dicot stem due to the formation of first vascular cambium and then cork cambium. Vascular cambium is responsible for the secondary growth in stellar region. Now, due to the activity of vascular cambium in the stellar region, there is some damage in extrastellar region. So, to heal that damage, there is formation of cork cambium that arises by the dedifferentiation of cortical cells. Cork cambium, also known as phylogen, produces sclerenchymatous cork cells on the outer side and parenchymatous secondary cortical cells on the inner side. Secondary cortical, also known as phyloderm, cork cells, also known as phellum, and cork cambium, also known as phylogen. Together, phylogen, phellum and phyloderm, together they are called as periderm. So this is the next important thing that you should know is what? Activity due to cork cambium. So cork cambium plus cork cells plus secondary cortex, they are together, they are known as periderm. I hope this entire concept of secondary growth in dicot root is clear, in dicot stem is clear. So what is done? There is normally dicot stem where vascular bundles are arranged in ring-like manner, conjoint open type. So the first step is formation of vascular cambium. The vascular cambium, which is circular in outline, develops due to the dedifferentiation of medullary rays. When medullary rays, they undergo dedifferentiation, they form what? Interfascicular cambium, thus completing the ring. This ring undergoes division, produces new cell, some inner, some outer. Inner cells, they get deposit to form secondary xylem, and some cells they deposit outside to form secondary phloem. Now, due to the deposition of such secondary tissues, what happens? The primary tissues, they start displacing. Primary xylem crushes the pith. Primary phloem damages the outer cells. Now, when such damage is being created, it has to be repaired also. Now comes the activity of Cork Cambium, which is produced by the dedifferentiation of cortical cells, general cortical cells. Now, these Cork Cambium, then they function and they produce some phloem cells and some phloderm cells, together known as periderm. And like this, whole secondary growth is complete. Clear? So ultimately, when the complete secondary growth is done in case of dicot stem, what will happen? Tell me. So first, let's try to draw all the layers in the dicot stem after 1 year secondary growth. 1 year secondary growth has been done, and now I am going to draw all the layers in its anatomy, starting from inner to outer. So first of all, it will have what? Pith in the center. Correct? Followed by pith, there will be primary xylem. Yes. After primary xylem, there will be first year secondary xylem. Tick? Secondary xylem. After secondary xylem, there will be vascular cambium. After vascular cambium, there will be secondary phloem. Right? After secondary phloem will come primary phloem. Then from primary phloem you come to see cortical cells. But here, now endodermal cells, pericycle cells, that is all okay. But soon after primary phloem, we'll start marking general cortex. So comes your general cortex, whose cells which got damaged are now repaired due to the formation of parenchymatous secondary cortex. And then there will be what? There will be phylogen or Cork Cambium, followed by epidermis, which is going to have some sclerenchymatous cork cells. Yes or no? So this will become the anatomy of dicot stem after first year of secondary growth. Let me label them. Centermost, pith, right? Followed by pith will be primary xylem. Followed by primary xylem will be what? Secondary xylem. After secondary xylem, there will be cambium. Near to cambium will be what? Near to cambium will be secondary phloem. Then after secondary phloem will come your primary phloem. Primary phloem will be followed by cortex cells that contain some healed up cortex called secondary cortex. Then will come Cork Cambium, and the topmost layer will be of repaired epidermis with cork cells. Clear? Now every year secondary growth takes place. There is more deposition of secondary xylem and secondary phloem. When there will be more deposition of secondary xylem and secondary phloem, the already existing tissues will start displacing. Already existing secondary xylem and primary phloem will crush towards the pith to give more space to newly formed secondary xylem. Similarly, already existing secondary phloem and primary phloem will move more apart so that new secondary phloem can be deposited, and this happens every year, and every year the thickness of the stem keeps on increasing. Clear to everybody? Yes. Now there is one concept of bark and stem, bark and lenticels. So what is bark? Bark represents all those tissues which are present outside to cambium. This all is bark. So bark is a technical term that includes all the tissues exterior to vascular cambium. That means bark includes phloem, cortex, epidermis and periderm. Correct? So what is bark? Bark means all tissues exterior to vascular cambium. I hope in the diagram you can clearly make out it. So that means what bark includes? See, that means bark now includes secondary phloem, primary phloem, cortex, secondary cortex, cork cambium and epidermis. So this cork cambium and secondary cortex can be called as periderm. Apart from periderm, bark also have both secondary and primary phloem. Clear to everybody what is bark? Will you be able to answer the question? Okay. Another term is called lenticels. Now, sometimes what happens, my dear students, when cork cambium is producing new cells, usually it is the activity of cork cambium to deposit cork cells, sclerenchymatous cork cells on the outer side and parenchymatous cells on the inner side. If sometimes on the outer side also, instead of sclerenchymatous, if parenchymatous cells get deposited, if instead of pink color, some brown colored, some parenchymatous cells also get deposited on the epidermal side, then these cells are known as complementary cells. These cells are known as what? These are known as complementary cells. What are complementary cells? The parenchymatous cells which are produced due to the activity of cork cambium on the outer side. These complementary cells, my dear students, are unable to heal up the injury of epidermis. Now, when cells deposition is taking place, but healing is not taking place, hence those epidermal regions, they remain opened, they remain damaged, and they form some lens-shaped openings now called as lenticels. And these lenticels, these openings then help in exchange of gases. They help in respiration and can also help in transpiration. Correct? So what are lenticels? Lenticels are, what are lenticels? Lenticels are the lens-shaped opening that are produced due to the deposition of parenchymatous complementary cells on the outer side of cork cambium. Parenchymatous complementary cells on the outer side. Clear? What is their role? Their role is to allow exchange of gases. I hope this concept of lenticels and bark, the two new terminologies which I have introduced, is clear. With this, secondary growth in dicot stem is done. Clear? Every year the same thing is going to happen and the thickness is going to increase. Coming to secondary growth in dicot root. Everything is same in case of dicot root. The same method of secondary growth takes place in dicot root, but with three major differences. Number one major difference is in how the vascular cambium is produced. When I talk about in dicot stem, 50% of the vascular cambium is already there in the form of intravascular. The remaining 50% only arises by the dedifferentiation of medullary rays, and then the complete circular ring is formed. However, in case of dicot roots, since the arrangement of vascular bundle is radial means closed, they don't have intravascular cambium since the beginning. So when the arrangement in dicot root is closed type, then how they will initiate the secondary growth? They don't have even that 50% of the cambium with them. So how will they initiate? So in case of dicot root, the pericycle cells, you remember? I was teaching you the anatomy. I told you that the cells of pericycle, they undergo dedifferentiation and form the complete vascular cambium. Correct? Yes or no? So in case of dicot root, the vascular cambium is completely secondary in origin, that is formed by the dedifferentiation of pericycle cells. Why? Because the arrangement in them is radial. Whereas in case of dicot stem, the vascular cambium is partially primary and partially secondary due to the presence of conjoint open type of arrangement. So this is the first difference on the basis of the origin of vascular cambium. In case of root, completely secondary. In case of stem, partially secondary, partially primary. Second difference is in their anatomy. What difference? In case of dicot root, vascular bundles are radial. In case of dicot stem, the vascular bundles are present in conjoint manner. Conjoint means they are open over there. Correct? Second difference. Third difference is, now in case of dicot stem, vascular bundles are present in ring-like manner. Therefore, the shape of vascular cambium is circular. But in case of dicot roots, due to the arrangement of vascular bundles in radial manner, hence their circular outline is not seen of vascular cambium. Rather, it is wavy in outline. So these are the only three differences which you have to remember. Rest whole process is same. So when it comes to anatomy of secondary growth of dicot root, the three things which you have to remember. Number one, in case of root, arrangement of vascular bundle is radial. If it is radial, that means it is always closed. Number 1 point. Number 2 point. The ring, the vascular cambial ring is completely secondary in origin. It's completely secondary in origin. How? It arises by the dedifferentiation of pericycle cells. This is the second difference. Third difference is the vascular cambial ring is wavy in outline rather than circular. Ring is wavy in outline. So these are the three only differences which you have to remember, and questions will be surely asked on the basis of all these 3 points. Rest entire secondary growth in dicot root is also same. The only difference is arrangement of vascular bundle, formation of vascular cambial ring, and shape of vascular cambial ring. Once you understood this, entire process is same. So with this, secondary growth is done. Now students, the last segment is to discuss some type of woods. Woods means wood is a common term used for secondary xylem. Right? Now I told you that every year secondary growth is taking place. Correct? And every year new deposition of secondary xylem and secondary phloem is occurring, and because of which every year the thickness of root and stem is increasing. So on the basis of every year growth in the thickness of root and shoot, we can count the age of the tree. We can tell the age of the tree. Now, before starting this topic of spring wood and autumn wood, let me tell you that the vascular cambial ring, that is secondary growth, takes place twice in a year. Once when it is spring season and second when it is autumn season, right? So definitely in spring season there is deposition of some secondary tissues, and another deposition of secondary tissues takes place during what? During autumn season, right? So the secondary tissues, the secondary xylem which is deposited during spring season is known as spring wood, and the ones which are deposited in the autumn season are known as autumn wood. One spring wood plus one autumn wood together makes one growth ring. So this one growth ring depicts the thickness. It's going to tell you that how your thickness of the stem has increased. Apart from this, by counting the number of growth ring, you can also tell the age of the tree, right? So there are two types of secondary xylem produced in a year due to the activity of cambium: spring wood and autumn wood. Spring wood means the secondary tissues which are developed during spring season, and autumn wood means the secondary tissues which are developed during autumn season. Now spring season is a good season, healthy season. So in spring season the cambial activity is very fast, and because of which there is high deposition of xylary elements. There are more amount of xylem deposited in spring wood, but during autumn season the activity of cambium is slightly slow, so there is less deposition of xylary tissues. So first point which you are going to write for spring wood and then autumn wood. Spring wood means the wood which is deposited during spring season. Autumn wood means the wood specifically means secondary xylem. We are talking in terms of secondary xylem. We are talking in terms of xylary elements. So wood means secondary xylem. Autumn wood means the secondary xylem deposited during autumn season. Now, second, in case of spring season, the activity of cambium is fast. As a result, more xylary elements are added. Here if I talk about in autumn season, cambium is slow, hence less deposition of xylary elements takes place. Third difference, in spring wood you will find that the vessels, in the xylary elements vessels, they have wide lumen due to less deposition of resin, tannins and lignin. So here you will write spring wood is better than autumn wood. The secondary xylem which is deposited during spring seasons are better for conduction as they have wider lumen. Why? Due to less deposition of chemicals like resins, tannins, lignin, etc. Right? But if I talk about autumn wood, it is not that much efficient in conduction. Why? Because they have narrower vessels. Why narrow vessels? Why their lumen become narrow? Because the lumens are occupied by high deposition of such chemicals like resins and tannins, the secondary metabolites produced by the plant. Now because in case of secondary wood, there is less deposition of such chemicals, such secondary metabolite, that is why their density will also be low. So they have low density and they appear to be lighter in color. Whereas autumn wood, if I talk about, in autumn wood, the density will be surely high density, and their color will also be dark due to more deposition of resin, tannins and other secondary metabolites. And now remember, one spring wood plus one autumn wood. This is actually the annual growth of the ring of the plant. Hence it is called as annual ring or growth ring. So number of annual rings is actually equivalent to the age of the tree. Right? So one spring wood plus one autumn wood is equal to one growth ring. Right? Yes or no? So this is how you can count the age of the tree, and this method of counting of the age of tree is known as dendrochronology. So this method of counting the age of the tree is known as dendrochronology. I hope entire secondary growth is now done. Now last type of wood which more you have to study is heartwood and sapwood. Now what is heartwood and what is sapwood? See, heartwood first of all is also known as duramen. So here I'll write duramen for heartwood, and sapwood is also known as alburnum. So I'm going to use these two terminologies so that you remember for longer time. Now my dear students, if I talk about what is alburnum and what is duramen, so just remember the diagram of secondary growth which I have shown you. Every year secondary growth takes place. Every year new secondary xylems are deposited. Now, if every year new secondary xylems are deposited, then already existing secondary xylem will keep on moving towards the pith. Yes or no? Suppose this is cambium. Here is secondary xylem, primary xylem and pith. This is cambium. First year growth only. Now second year more secondary xylem will come. Now if again secondary xylem will come, it will come over here. Now due to deposition of new secondary xylem, the already secondary xylem will crush towards the pith. One more growth will again next year, new secondary xylem. So now the last year placed secondary xylem will again move towards the pith. So like this keeps on happening. Every year new secondary xylem is deposited towards the cambium, because of which the already existing secondary xylem they had to displace and they have to move towards the pith. So what happens? The newly formed secondary xylem are efficient in conduction, they have less deposition of resins and tannins, their density is light, and they are called as sapwood or alburnum. Whereas those secondary xylem which have gone inside the pith, which have occupied the centermost region of the plant body and which are not now responsible for conduction of water and mineral, as they have heavy deposition of resins and tannins, their vessels have become completely narrow, they're high in density, dark in color, that is known as heartwood or duramen. Correct? So what is duramen? What is heartwood? Heartwood comprises of secondary xylem which is near to the core. They are high in density. Why? Because they have heavy deposition of resins and tannins, because of which they appear dark in color, and hence they cannot help in conduction because their vessels have become extremely narrow, so they cannot help in conduction. Rather, now they start only providing mechanical support. Now, talking about alburnum or sapwood, these are the newly formed secondary xylem which are present close to the cambium. So these comprises of newly formed secondary xylem close to cambium. Right? Close to cambium. Because they are recently formed. They are recently formed. Hence, there is less deposition of resins and tannins. They show less deposition. If they're less deposition, then surely their density will also be less. And coloration will also be lighter. Right? And since they have less deposition of such secondary metabolite, their vessels will be broader and will help in conduction of water and mineral. So this is the difference between heartwood and sapwood, springwood and autumnwood, which all are the types of secondary xylem on the basis of secondary growth. With this, finally the chapter is done. Question part time. So first question says innermost layer of cortex is known as. So what is the innermost layer of the cortex? You know it is pericycle, no. Pith, no. Hypodermis is present outer to cortex, so no. Hence the answer of this question becomes C, endodermis. The innermost layer of the cortex is endodermis. Hope you know this. I have already told you while teaching the chapter. Now, moving ahead, second question. Vascular bundles with cambium are known as. Vascular bundles that have cambium in between. So, this type of vascular bundles are open type. That means they can undergo secondary growth. Correct? So the correct answer will be what? Radial always closed, wrong, none of the above wrong. Answer becomes B. Hope you have understood, and with this we finally end our today's session of anatomy in flowering plants. Very important, very important. Why? Because so many questions are asked from this section in your NEET examination. My dear students, it's a high time that you all start preparing for your NEET. If you have any doubts, any queries, you can ask in the comment box. But please go through the lecture properly, and please make sure you share it with your friends and everybody, so that if you are taking the benefit, some other person can also take that benefit. Any doubts, any queries, you can anytime post on the comment and I'll surely reply. But apart from questions and queries, even if you have any other problems, then too you can share. If I have the ample amount of time, I'll surely reply to all your queries. And with this, I would like to say thank you. Bye-bye. Take care. See you in the next session. Keep working hard. Keep connected to Physics Wallah. Bye-bye, everyone.